Okay, it's already 5 past 10, so I think we'll get started. And apologies for the tiny late start. So good morning, everyone, and welcome to GCFI74 Day 4. We're in for some exciting presentations and panel discussions today. And we're going to get things started off today with the invasive species session. So I'm going to hand over to our moderator for today, Alex Fogg. So please, Alex, over to you.
Thanks, Fadila. Good morning, GCFI. Unfortunately, we aren't in person, but fortunately, we're able to have this meeting online, and this platform is fantastic. We have a number of awesome, quick talks this morning. Hopefully, everyone's been able to go look at the recordings already. But we're going to start with Dr. Holden Harris, who's a postdoc researcher associate at the University of Florida, and he's going to be talking about the bioeconomic paradox of the commercial lionfish fishery. So Holden, you have 3 minutes. You can go ahead and get started.
Thank you, Alex. So my co-authors in this are Will Patterson, Rob Ahrens, Mike Allen, Dave Shigeris, and Sherry Larkin. And I think this is really interesting in that market-based harvest of invasive species offers a really compelling approach to control their abundances and mitigate their impacts without using public resources. I think this appeal can be exemplified in the recent invasivore movement in things like invasive species cookbooks. However, there's an economic paradox, right, in that achieving the goal of over-harvesting the unwanted species will diminish catch rates and make their harvest unprofitable. So we examined this paradox with bioeconomic models of the commercial spearfishing fishery for invasive lionfish in the northern Gulf of Mexico. Specifically, we examined whether open access equilibrium commercial harvest will be high enough to control lionfish. If not, what types and how much additional support may be necessary. To do so, we quantify biological and economic characteristics of the fishery with a variety of approaches and data sources. We then use these parameters to estimate the open access equilibrium point solutions and develop time dynamic simulations of the fishery. One of the findings from these models is that using different methodological approaches estimated different biological parameters and really provided a range of intrinsic growth rate estimates for lionfish. So population net growth models with these are shown in figure 2. And in part, this indicates that there's considerable uncertainty around forecasting lionfish populations, particularly given their recent population declines. Overall, our models indicated that lionfish populations were quite robust to fishing under a variety of different scenarios. So this means that reducing lionfish biomass was difficult, but it also indicated that a stable open access fishery could develop. So at open access equilibrium, we predicted that the fishery would reduce the lionfish stock biomass to about half of its regional carrying capacity. A fishing effort at this open access equilibrium levels would be about 7% less than which we predicted to produce maximum sustainable yield, or MSY. This solution point is shown in figure 3 as the intersection of the sustainable revenue curve and the cost function. And you can see there that to the left of the apex of the sustainable revenue curve, which is where MSY would occur. However, we also were able to consider hypothetical increases in the price of lionfish or decreases in the cost of fishing, and this would be able to reduce lionfish biomass below BMSY. Figure 4 shows that to reach the open access equilibrium solutions, current yield would need to increase about approximately 4 times, an effort by approximately 8 times higher than the peak levels that we observed in 2017. It's also notable that not all of our time dynamic simulations reach equilibrium. Some of the scenarios projected cyclical boom bust fisheries whereby high levels of yield would crash the stock biomass, then efforts would subsequently crash, and the stock would recover and the cycle would repeat. So I think really the data limitations for these models likely preclude them to be able to be used tactically. And unfortunately, I don't think they're robust enough to really set specific targets to hang your hat on. However, I really think they may offer valuable strategic guidance for how management can help support market-based control efforts for lionfish and other invasive species. So models like this can test key assumptions. For example, that invasive species removal will have desired outcomes, by explicitly considering how density-dependent mechanisms, the same ones that lead to sustainable fisheries, are concurrently problematic for attempts to control invasive species via commercial exploitation. I'll also argue that these efforts to explicitly parameterize these models can help set objective targets and provide metrics for adaptive management. So please reach out to me if you have any questions, if you'd like to dive deeper into this or really nerd out about the modeling, we can get in deep. Our paper was recently submitted to Biological Invasions, and I'm happy to share that manuscript and discuss it further. Thank you.
Awesome. Thank you very much, Holden. Great summary. Again, if there's any questions, please wait until we make it through all of these summaries before, I guess, asking all the questions you may have. The next talk is going to be Ms. Stephanie Green, who is an assistant professor at the University of Alberta. She's going to be talking about broad-scale acoustic telemetry. It reveals long-distance movements and larger home ranges for invasive lionfish on the Atlantic coral reefs. Go ahead, Stephanie.
Good morning, everyone. It's a pleasure to be back at GCFI. It's been several years for me, but pleasure to see you all here, at least virtually and hopefully in person in future times. I had a slide, and Alex, I'm not seeing it up, but maybe my computer's just slow. Aha, there we go. Okay. So briefly, this project is a collaboration between scientists at the University of the Virgin Islands, National Park Service, Reef Environmental Education Foundation, and others, funded by Virgin Islands EPSCoR grant from the National Science Foundation, and was initiated through graduate work led by Elizabeth Smith in Buck Island Reef National Monument in St. Croix. And what we did there was take advantage of the large-scale acoustic array that exists around Buck Island Reef National Monument to focus that ability on tracking invasive lionfish. Previous work done on the species has suggested fairly small home ranges and low movements, but one of the challenges is that often individual fish exit the array or leave the study area. And so we're not certain exactly that we're capturing the full range of movements undertaken by the species, but it's really important for management in terms of identifying the scale of culling that might be needed, and also understanding better drivers of recolonization following control. So what we did was tag 35 lionfish with acoustic receivers, and a subset of those had accelerometers, so we could get vertical movement as well. And what we found by tracking these individuals over 14 months in a 35 kilometer squared array is that lionfish actually move more than we thought. Maximum distance that we saw one individual move was 10 kilometers in just 10 days. And many of the long distance movements that we saw, which were between 2 and 10 kilometers over that study period. Several individuals made directional movements from a tagging area that was in shallow patch reefs, north, west, and northeast to the shelf edge to deeper water. And this was consistent among many individuals that did make long distance movements. We also found that overall home range in lionfish is at least 3 to up to many times larger than previously suggested by other work, with a mean of about 121,000 square meters or 12.1 hectares. We were anticipating that we might see some interesting patterns with lunar phase or perhaps with body size, but we actually didn't find anything clear in terms of monthly cycles or seasonal cycles in terms of movement or long distance migrations. We did find, of course, as other studies have found, patterns of movement that fit with daily cycles of light and dark. So for example, high activity rates at dawn and dusk that likely correspond with feeding patterns, and lower activity at night and during the day, which makes a lot of sense and gives us more confidence in our approach. But sort of stepping back and looking at what a study like this might mean, we think it has some implications for advice for culling programs which are happening. And that is that perhaps the size of areas to be culled, if they're greater than 10 hectares in sort of overall area, they might benefit from reduced recolonization, because that's sort of achieving the average home range size of lionfish seen in the study, or sites that are disconnected from other habitats by about 200 meters or so, which is sort of the mean radius of the home range that we identified, might also benefit from reduced recolonization. So as programs are planning where to cull and when, looking at perhaps for these site characteristics and thinking about logistics in this way might help to increase the efficacy and benefits in terms of reducing that recolonization rate that we have seen from many studies that study this problem. Okay, I should also just say before I hand it over that this paper is published in Marine Ecology Progress Series. It is open access. I was going to put the full link with all the crazy numbers and letters, but I also just have posted it to our lab website, which might be easier for you to jot down and find, or you can just search for the title of this talk. Thanks very much, everyone.
Awesome. Thank you very much, Stephanie. That was great. Our next talk is going to be by Kaylin Clements, and she is a doctoral candidate in human dimensions of natural resources at Colorado State University. She'll be talking about regional synthesis of invasive lionfish citizen science programs in the tropical Western Atlantic. Go ahead, Kaylin.
Thanks, Alex. I'm realizing after seeing the first two presentations that I could have put more on my slide, but I'm glad that my co-authors, many of whom are on this call, are featured very clearly here. Like Alex said, I'm Kaylin Clements, and I have the pleasure today to tell you about lionfish citizen science in the Western Atlantic. And this research has been written as a paper currently in review, and the new title is here, The Role of Citizen Science in the Research and Management of Invasive Lionfish Across the Western Atlantic. As you all know, the lionfish invasion is widespread. It's difficult to control and to research, and it appears beyond the capacity of just natural resource organizations. In fact, in our study, we found the two primary obstacles organizations working on lionfish face are funding and staffing. So how much do we rely on citizen scientists for lionfish management and research? To answer this question, we distributed a survey in Spanish and English and received responses from 71 organizations that work in all but 4 countries in the invaded Western Atlantic. And I can give you more information about our methods. We also explored 5 case studies, including research and control efforts by Flower Garden Bank's National Marine Sanctuary, Reef, Stanapa Bonaire, Reef CI, and Lionfish University. And first off, in the survey, we asked respondents about their involvement in 21 different approaches that ran the gamut of strategies for lionfish management. And then they ranked which approaches they saw as the most impactful in a subjective way. And the top 6 were organized culling and removal, public education outreach or awareness raising, tournaments and derbies, promoting consumption of lionfish, and data collection. And it just so happens that these 5 approaches were also the ones that organizations most commonly involved citizen scientists in. I should also add that organizations tend to implement multiple approaches at once and engage citizen scientists in multiple approaches. For instance, derbies and organized culling often facilitate lionfish removal and data collection at the same time, as you can see here in this picture. In fact, there's somebody in the background also cooking up lionfish for a tasting. The majority of respondents did not engage citizen scientists, their organizations didn't, but among the organizations who did, 60% indicated they would be unable to undertake their lionfish management work without them. And when we asked respondents to rate the level of importance of citizen scientists to scientific findings, data collection, and lionfish management efforts, the majority saw them as moderately to extremely important in all 3 areas. So citizen scientists are pretty crucial, but we wanted to know what tasks they're involved in more specifically. And we found that of all the steps of the scientific process, members of the public are by far primarily collecting lionfish data. And this data has contributed to peer-reviewed scientific publications, it's informed management, contributed to government agency research, and it's informed policy. And as important as data collection is, however, we suggest that it would be wise to engage citizen scientists in more elements of the scientific process. Research has shown that engagement in more parts of the process, and especially in impactful activities that lead to clear outcomes, increases citizen scientists' involvement in the overall cause. We see this with engagement across multiple lionfish management approaches, such as engagement with removal of lionfish, which is a very clear outcome and is impactful, but we see an opportunity for organizations to engage citizen scientists in more steps in the scientific process when possible. Thanks so much. That's my sprint. That's my 3-minute sprint.
Very good. Thank you very much. Good talk. I look forward to asking some questions after this. Next talk is going to be by Alexandria Davis, and she is an early career professional at the University of Alberta. She's going to be talking about multiple drivers of invasive lionfish culling efficiency in marine protected areas.
All right, thank you. Hi. Yeah, so I'm going to be talking about a paper that Steph Green and I just recently published with a lot of really awesome co-authors from a bunch of different regions in the Caribbean and the invaded range of lionfish, and that many of you on this call helped collect data for. So thank you for all your effort that went into creating this paper. But the premise behind this paper was to look at 3 different regions where lionfish removal was happening and take characteristics of the invader and the system and the way that we're moving lionfish in order to create some sort of measure of efficacy of what leads to efficient lionfish removals. And so, as many of you may know, we went out to the Flower Garden Banks National Marine Sanctuary, Buck Island Reef National Monument, and then, Biscayne National Park, and where's my last one? Florida Keys National Marine Sanctuary, and collected a ton of data on lionfish removal. And since you all know, most of the removals happen with spears, that allows us to collect a really rich data set of who's doing the removal, how much time it takes to do the removal, where the lionfish was on the reef, what size it was it hanging out with any other lionfish, and what was its behavior and time of day. And so using all of these traits of both the environment, the habitat, the remover, and the invader, we created models to see if we can estimate what leads to the likelihood of removing a lionfish, higher densities of removal, and then also the time it takes to remove lionfish. And so the slide I have up here is just kind of the screenshot of like what saves time for management. And I thought that one was the most interesting result. And so the results are really intuitive for anybody who does lionfish removals, but it's really nice to see it done quantitatively and presented here, where we see higher densities of lionfish, and larger lionfish are easier to remove, so they take less time. Remover experience does make a difference. Reef size actually contributes a little bit to removal effort, and then time of day is really important. And so kind of the take home from that is that removing lionfish at dawn and dusk often takes 1 minute less per fish across all experience levels. And then adding experience, so folks who have either a lot of diving experience or have had training in lionfish removals before they came out to participate in this project also saved a minute per fish. And so, as you know, time underwater is precious. And so going out at dawn and dusk with your experienced removers can often save you 20 minutes per dive, which if you have a lot of area that you need to cover and multiple reefs that you need to remove them, really adds up for both time spent on the water and time spent underwater. And for me, the really interesting part about this is that these are metrics that managers can account for without ever getting in the water, without ever setting foot on a boat. And coming to the next step of what we're doing is looking at density and recolonization. Those will then be able to be added into your management plan. And like I said, this paper just came out in Conservation Science and Practice, and I can throw the actual link to that instead of just the short citation into the chat if anybody's interested. Thank you.
Great. Thank you very much. Really awesome to see some of this data presented. It's a lot of diving associated with that. A lot of fish died too. All right, next up is Eileen Ullman. For those of you who don't know, she's all the way over in the Mediterranean She's a marine conservationist with Marcy Marine Conservation, and she's going to be talking about policy recommendations for the Mediterranean lionfish invasion based on lessons from the Western Atlantic. Go for it.
Thanks, Alex. This is my first GCFI event, so I wanted to thank everyone for the invitation, first of all. Before academia, I did spend quite a few years working in the Caribbean, and my last job was at Reef CI in Belize in the late 2000s. I spent 2 years there as a dive instructor. And I just missed the lionfish invasion there because it started just a couple of years after I left. But I had been following it very closely since I left, trying to stay updated on their predators and culling initiatives. So fast forward 10 years, now I'm working in Turkey where I am supposed to design the best incentive mechanism for lionfish control over here. We had our first lionfish record quite a long time ago in about 2013, with abundances increasing only over the last few years. Last year in 2020, the UNDP Turkey organized an East-West lionfish knowledge exchange where many American colleagues, such as Alex and Holden and Steve, provided very detailed information and helpful advice about best practices for control over the Mediterranean lionfish invasion. After this meeting, it became clearly apparent that the most pressing issues over here is that policy currently prohibits scuba diving and spearfishing in the most affected countries, such as Greece, Turkey, Israel, and Lebanon. And after realizing the high level of control culling has provided in the Western Atlantic invasion, it became clear that the most important next step is to influence policy changes to permit culling over here in the Eastern Mediterranean. So with very helpful guidance from many of the people on this panel today, Holden, Alex, Fadila, Steve, Lionfish University, among many others, we started writing a policy paper with the following goals. First, to show the demonstrated examples that culling does work for localized control, because that is not known yet here. Second, to show that most Western Atlantic states have altered policy in some way, and many options do exist for amendments to allow for culling of lionfish with scuba gear, whether it's permitted through NGOs, through divers, or through derby events, for example. We also explain what has worked best for lionfish management. For example, how derby events, as Kaylin was saying, are often combined with outreach and tasting events, and what is not advised, like why bounties did not work and why you should not feed lionfish to potential predators. So this has been a really beautiful collaborative effort where your best practices and tons of work over there are being put to good use in the Mediterranean. So I thank all the people who have and who are still contributing to this research, and look forward to many more east to west fruitful collaborations. So our paper, we hope to submit it in a month or two, but if anyone has any questions, we would love to hear them. Thanks for your time.
Awesome. Thank you very much. Great talk. And last but not least is Melanie Esch, and she is a PhD candidate at Florida International University. She'll be talking about predicting the potential distribution of a new Caribbean invader using a species distribution model.
Hi, everyone. Good morning. Are you able to hear me? Hopefully. All right, great. So as Alex mentioned, last summary, not about lionfish. Hopefully you all are game for hearing about something new. So I'm hoping that many of you have seen this fish or are at least aware of this fish that you see in the photo because of the regional focus of this conference. But for those of you who aren't, this is the regal damselfish, Neopomacentrus cyanimus. This is a species that is native to the Indo-Pacific, and it was first observed over here in the Caribbean, specifically the Gulf of Mexico in 2013 in Veracruz, Mexico. And since then, they have been able to expand their distribution all the way east to the Yucatan Peninsula, as well as along the US coastline. And their current invasion front is in the Florida Panhandle near Panama City Beach. So the potential range expansion and entire distribution of the species is relatively unknown in the Greater Caribbean and was the goal of my research. And so to predict that potential distribution, I have created a species distribution model using both native and invasive data for this species. So in the map here, you can see this is a habitat suitability map, which is the output of my model, where that salmon color is going to represent highly suitable habitat for this species, and the dark blue is going to represent less suitable habitat. So with this map, I do want to point out a few key findings here or highlights. And the first is that this species is going to hug the coastline where the water is relatively shallow. Makes a lot of sense. They usually are found on reefs or artificial structures where, again, it's going to be relatively shallow. The second is that this species is likely to arrive to South Florida. And on the map, we can see that there is highly suitable habitat all along the west coast of Florida. But we haven't really seen a range expansion in this direction in the past couple of years. And I suspect that is due to the lack of natural reefs. So I kind of think that they are having to use artificial structures as stepping stones, which is kind of slowing their advancement in this direction. And the last point I wanted to make is that it does appear that there is highly suitable habitat in a lot of the Northern Caribbean, especially around the Bahamian Islands. And so if they are able to make it into South Florida and the Florida Keys, this could be a good launching point for this species to get into some of those other Caribbean islands. And this is also something that was seen with the invasive lionfish, where there was a lag after they were first observed in Florida. But once they did make it into the Bahamas, there was a big explosion of those populations. So if this species, again, is able to make it to the Florida Keys, we might see a similar pattern with that distribution expansion, as well as increased abundance in those areas. So hopefully this model can be useful for management to try and slow the spread of the species and do some targeted monitoring to see how they advance into new areas. Thanks, everyone.
Great, thank you very much. At this point, we're going to open it up for some questions. I see some folks put some questions in the chats and the Q&A tab. I think either works, correct, Fadila?
That's right.
Okay. Let's see here. All right, we'll start with a question for Steph Green. Your work, as well as Kristen's recent telemetry study in the Northern Gulf of Mexico, both conclude that lionfish movement appears to be higher than what was previously estimated. Why do you think the more recent assessments are finding broader home ranges? Could lionfish be possibly moving more now that they've become established, or is this more the recent uses, or is this because the more recent uses of larger telemetry arrays and the previous arrays were too small to capture these home ranges?
Yeah, thanks for the question. I think it's very likely to be our focus on larger scale and longer term tracking that's revealing the possible extent of movement. I think, one thing to highlight from our study that's been seen in others is that home range and movement is quite variable among individuals, and we're still trying to understand the drivers of that variation. It didn't appear to be body size, at least for fish over 18 centimeters, which are already, reproductively mature, and it didn't appear to be lunar phases or seasons in a big way in our study. But I think it's likely that we are better able to capture the full extent of that variation now with some of the studies that are coming out, rather than a change over time, I would suspect.
Awesome. And to follow up on that, did you see, you said you had pressure tags implanted?
Yeah.
Did you see vertical movement?
We unfortunately did not have many individuals with that survived a long time within the study system with that sort of larger tag. And so we weren't able to capture a ton of vertical movements. And I actually, I'm gonna mute myself and look into that, and maybe I'll ping you when I can remember exactly what we did see, because I remember we didn't actually end up featuring that too much in the paper.
Okay, great, thanks. Moving on to Holden. What would happen if lionfish populations get more profitable throughout the years because entrepreneurs want to keep landings high for selling the fillets?
Yeah, I think that's a really interesting question. Thanks, Alfonso. I think it likely depends on a lot of things. So again, these models do try to forecast the future, but there's a lot of uncertainty based on the biology. But it could lead to some potentially interesting dynamics. What we've forecasted with the current model is that we're actually below maximum sustained yield, but it kind of just creates like a stable fishery. If the price of lionfish keeps going up higher, which is something that we'd want to encourage, either potentially with public subsidies or like with marketing efforts to increase the price of lionfish, or even value-added products, like for instance, Eileen's work to create jewelry from lionfish could increase the price of lionfish. What we expect is effort's going to increase. If it does get too high, what you could actually get is unstable dynamics in the fishery, where basically a bunch of people enter the fishery, overfish lionfish, and then they leave the fishery, and you get these kind of boom bust cycles.
Awesome. Thanks, Holden. Do you suspect that, the fishery may not be contributing to the declines that we saw? I mean, up here in the Northern Gulf of Mexico, as you know, we saw an outbreak of some sort of disease. Do you think that that was lethal and caused the declines that we saw?
I mean, that's my guess, that we saw that disease. There's that paper that we worked on that said that relative condition, especially for females, was lower, and then we had a recruitment failure the next year. And so we think that the disease was kind of attributed to those declines. It's really hard to disentangle how much the fishery caused the declines, because we can see that it does drive populations down, and then how much natural mortality is causing the declines.
Awesome. Thank you.
That's the way of saying I don't know.
Yeah, very non-committal. Okay, I'll leave Stephanie alone for a minute. We'll go to Alex. Do you think removal efforts are worth?
Maybe worth it.
Oh, okay. Are removal efforts worth it because greater lionfish abundance are in mesophotic reefs beyond 40 meters?
That's the question with all invasive species. Is it worth it to spend all our time doing removals if we can't effectively remove all areas? I think yes. One of the things I'm also interested in, kind of following up on this paper, is being able to compare some of these non-human centric removal efforts. And so like the traps that people are coming up with, or some of the remotely operated vehicles, and how does that, can we quantify the time and effort put in the same way as we do for what we do when we have all the data on humans going out and removing them? And I don't think it's going to be a direct one-to-one comparison, but I think it would be worth it to see how much more efficient some of these remotely used removal efforts are compared to us going out and doing it by hand, because obviously for mesophotic reefs, it's not worth our while to go down as divers, do removals. And so some of these other options are really, really amazing for getting to that area of the invaded range that we just can't reach on a daily basis. So I definitely think it's worth it. Reducing the populations in any place will reduce the population. And so but also figuring out whether or not those lionfish at the in the mesophotic depths are like a source for the fish that we're seeing up there. And so there's a lot that could go into whether or not the removals are just kind of like, we're just gonna have to go out all the time, and most of you all know this, is like there's a lot of complexity, but I think they're definitely worth it. And I think figuring out the efficiency of the non-human removals will also be really, really helpful in figuring out how often we have to go out to do removals by hand.
Awesome. Thanks. I agree too. It's always worth it to get out there and harvest lionfish, whether they're shallow or deep. And Alfonso, you're busy. So we'll go to Melanie. Do you consider this damselfish to be invasive in the general term of the definition?
All right. So most people are on agreement that in order for a species to be invasive, they have to be showing some type of negative impact in their new environment, and impacts of this damselfish in the Western Atlantic have not yet been quantified. So I suppose the easiest answer is just to say no, they are not yet invasive, and use just more general terms that I often use in describing the species as an alien species rather than invasive. However, I will note that when you're talking to the general public and also non-scientists, often it's easier to refer to these species as invasive so that they're more understanding of what exactly you're talking about and have something more relatable to consider. So no, they're not potentially invasive or even going to be invasive in this range. We don't know yet.
Okay, awesome. Thank you. And while I have you up here in the Northern Gulf of Mexico, as you indicated, we have a lot of these regals, especially late in the season, late in the summer season when the water's pretty warm. Do you suspect that the cooler temperatures are probably going to keep this fish at bay up here in the Northern Gulf, or do you think they'll be able to overwinter?
Yeah, so I've had some conflicting data for this. So temperature actually wasn't one of the key drivers for my species distribution model. It fell a little bit short, although it did play a role. So it was showing that they do prefer warmer temperatures, but I'm currently also doing some experiments to try and determine their thermal tolerance in the lab. And what I'm seeing is that they can actually survive in pretty cool temperatures that they would experience in the Northern Caribbean, but it could be that they're really only thriving in the deeper water habitats in the Northern Gulf of Mexico that don't have quite as a dramatic temperature shift in the winter months. And that might be why we're seeing such an abundance during the summer months. And maybe a little bit less during the cooler water days.
Awesome. Thank you. Stephanie, are you ready for one?
Yes.
Okay. One hypothesis for the continued colonization of lionfish on reefs is that they move up from deeper areas that are not accessible to divers. Do you see any evidence of this in your work? And how does this jive with your limited movement home range data?
Yeah, thank you for the question. So we tagged fish for logistical purposes, not using rebreathers for the study. We tagged fish on more shallow patch reefs south of Buck Island in the monument. So at sites about 40 to 50 feet deep. And so because fish were tagged there, we were then tracking their movements from that initial point to different locations within the array. So because we didn't tag fish sort of off the shelf in mesophotic areas, we weren't able to then look for movement from that area into shallower water. But given what we know about the general movement patterns and movement ability, I wouldn't be surprised if there is recolonization, especially if those areas have been culled less frequently and we might have higher densities. We know that lionfish are moving between areas of suitable habitat, and so I can't imagine why we wouldn't see recolonization from areas that are sort of in mesophotic zones if there's shallower reef adjacent to that. In terms of why we might see these movements, I still am not clear. We know that there are sort of individuals that will leave an area and not come back to it. We're not sort of seeing cycles of movement. And so I think there's still a fair bit of work to be done on some of the drivers of movement. To answer, Alex, the question originally about vertical movement, unfortunately, we had 6 fish tagged with pressure sensors and accelerometers, and 5 of them were mortality events, leaving 1 fish. It's a very sad day for the project. I do believe that we saw vertical movement, but I don't believe there was any clear pattern in terms of drivers, in terms of time of day or season with that. I can look back into it.
Awesome.
Yeah.
No, that thank you. I mean, that's been a question I've had for a long time is this vertical movement happening, whether it be for reproduction or other purposes, but we can't seem to capture it on video or with tags or anything. So thank you.
just one last thing I should say, I believe that Rick Nemeth at UVI and collaborators from a couple of other institutions have done some mesophotic tagging work. I'm not sure if that has been published yet, but I believe that would be coming out soon as well.
Very cool. All right, Eileen, have you noticed the presence of volitans in the Mediterranean? Because it's mostly miles there, right?
Yes, there have been no affirmed cases yet of volitans. There was one study that said it was here, but there was no proof of that, and they didn't deposit the genetic code anywhere. So just miles as far as we know.
Okay.
But while I have you here, if anyone needs a research project, I just wanted to mention that in the Mediterranean, we have about 80 invasive species in Turkey and about 500 alien species. So if anyone needs a research topic, you can come help us over here. We would welcome you willingly. Thanks.
Thank you. All right, back to Stephanie again. What was the size of the lionfish that traveled 10 kilometers in 10 days? And a follow-up to that is, do you think it might have been a shark or something that may have eaten the lionfish?
Now you're testing my memory for the details at this time of the morning.
Dig
I
deep.
will get back to you on that one about the body size. They were all over 18 centimeters that were tagged, so between 18 and 34. So I'll figure that out for you. But so we investigated. One of the big challenges with this was to kind of interrogate the data set and remove detections that we thought might be from drop tags or predation events or mortality and other sources of mortality. And so we actually did a couple of different scenario analyses in our calculations, including and excluding data that were actually including and excluding data that were sort of on the lower movement end, because lionfish are quite sedentary sometimes. We didn't want to exclude fish because they might have died, when really it might just be that they're sticking to one or a few locations. On the other side of that, we also set sort of maximum rates of movement that we thought might indicate a predation event, and that tag being carried with a predator. And the fish that moved this 10 kilometers in 10 days was below that sort of limit that we set. So it seems unlikely, because there were stops along the way, that it was a predation event.
Very cool. Thank you. All right, back to Melanie. Your habitat models suggest that the Yucatan and off Costa Rica are a great habitat for damselfish. Does your modeling give any sort of timeline of when this might happen? And are there enough stepping stones between the Florida Panhandle and these regions?
So unfortunately, my models do not have any sort of timeline to them. It would be amazing if they could. However, one thing that I didn't get to mention during the summary, but if you watched my full talk, you would have seen, is that there was actually a second introduction point observed in the Western Atlantic. So in Trinidad, this species was also observed in, I believe it was the summer of 2019. And there have also been individuals observed around the island of Aruba, which it's suspected that that was expansion from that population in Trinidad. So, you know, in a lot of ways, we kind of have these two populations now that are going to start kind of suffocating, I guess, the rest of the greater Caribbean islands that don't have this species yet. So they kind of have multiple angles of attack to try and reach some of these areas. But in terms of coming off of the Yucatan into areas like Belize, the currents are moving in the opposite direction. So it is going to likely take them a little bit longer to find their way into these areas, as a lot of their larvae, while they're at the whim of the ocean currents, are going to be pushed back into the Gulf of Mexico rather than further south into Mesoamerican countries.
Okay, and do you think there's going to be some interaction between lionfish and these damsels?
Great question. It's also one that I have had and am trying to do some research on. I do think that there will be. They come from a similar native range. And so it's kind of a cool idea, thinking that, you know, these damselfish might be better able to avoid lionfish than some native damselfish species are able to, due to innate recognition. So that is something that I am working on and am trying to try and discover if there is interaction and potential kind of a superior competition effect due to their innate recognition of lionfish.
Okay, and could you go over the current distribution of the regal damsel again, and repeat the time frame from Veracruz discovery? Because this was from Ron Hill and he missed it.
Yeah, sure. So they were first observed near Veracruz in 2013. So it has been several years since then. They've expanded north into the US coastline, and their current invasion front in that aspect is near Panama City Beach. And then we have individuals all along the Yucatan Peninsula. And then again, we have that second site of introduction around Trinidad and also individuals in Aruba.
Awesome. I will leave you alone for a little while. On to Holden. If I understand your methods correctly, stiffness relates to fishers' response to the price of lionfish, correct? And is it difficult to predict? If that's correct, then what type of data would you need to better inform that variability?
Yeah, so that's a great question. I know Kaelin's pretty familiar with this work. So the stiffness parameter is, to give a little bit of background, it's basically how strongly the harvesters respond to profits. And that informs those time dynamic models. The open access equilibrium models don't have stiffness in there because it assumes the fishery is at equilibrium. But what we saw is that we ended up not being able to estimate stiffness, and we did see different effects when we modeled high stiffness versus low stiffness. And it really says that's an important thing. And so you bring up a really good limitation and point is that we don't have that. So there's two ways you can estimate stiffness. If you have a long time series of catch data, you can get a robust estimate based on what we see from basically historically fishing effort following what we estimate profit to be. We had a limited time series of data, even though lionfish are, one of the best studied marine invasive species in the world. That's far less studied than, for instance, like a formal stock assessment. And we weren't able to estimate a robust parameter for stiffness. The other way is social research. Of course, you can go out and ask fishermen their basically willingness to fish based on different conditions. And I think that really suggests that if you wanted to make more accurate forecasts, that's what I'd recommend researchers do. And anyone that's interested in that, I think that would be a really good study and follow up to this. And I think it's especially interesting because lionfish are an invasive species. So even though we're modeling commercial harvesters, not recreational harvesters, so we're assuming that they're profit driven. Commercial harvesters are also aware of lionfish and they don't like them. They interfere with their other species. And so they might harvest them even regardless. So that stiffness might be different for an invasive species as it would for like red snapper, or gag grouper, or another commercial species.
Thanks, Holden. All right, we're going back to Melanie again. This one's from Kelly down in Trinidad and Tobago. So they've already made it there very recently. Your model does not show them moving further up the West Indies. Why not? Also, I don't notice, but did it show them reaching the ABC Islands, Aruba, Bonaire, Curacao, as they have been reported there, unconfirmed though? Currents from here run either north or west. Also, there are plenty of natural hard bottom reefs in the Big Bend area of Florida. She used to work up there extensively, so she's happy to provide more information about this.
Hi, Kelly. Thanks so much. Yeah, I've had a few conversations about your work with Ross, so I'd be happy to catch up with you. So the map that I showed in my both my long talk and the summary was covering a regional scale. So a lot of those islands are teeny tiny on my map, and you would really have to zoom in to see exactly what the habitat suitability is around those islands. But there is suitable habitat. As you're probably aware, a lot of the reef shelf is quite narrow along a lot of those islands. So the actual habitat that will be suitable is very slim around several of those islands in the West Indies. but there is suitable habitat there for them. And I think that is also true with the ABC islands that you mentioned. It just, again, is a little bit hard to visualize in the map that I displayed, but I would be happy to show some more images with you and even talk about this work a little bit more.
Great, thank you. This one's for Holden from Stephanie. There we go. Would boom and bust dynamics be acceptable in a multi-species fisheries context where lionfish are added to harvest strategies, variability over time as catch of other species restricted or lionfish become abundant?
I think, again, I don't know, and I think we'll have to see, but I think so in that I think the lionfish fishery might be kind of different and that people might not be entering the fishery just for lionfish. It's whether or not they're already doing other commercial spearfishing and then also harvest lionfish or not. So I think that, for instance, that might mitigate some of what we like numerically modeled as kind of boom and bust fishing using kind of these really traditional fisheries methods, which are built more for like trapping, trap fisheries where you have significant capital investment. Lionfish fisheries, I think, are more liquid of people being able to move in and out of a commercial fishery in sense of basically whether or not they take a zookeeper when they're diving or not.
Thanks, Holden. I got a quick one for Alex. Is the minimum amount of time for your work to harvest a lionfish, was it 1 minute or did you go sub 1 minute?
Sorry, I can't find my buttons.
Okay.
There were a lot of sub 1 minute. So quite a few of you are very fast at catching lionfish, and there were a lot of times that were reported basically near zero from time to sighting the lionfish to time to actually spearing it. And so it was a very non-normal distribution where we had a lot of really fast removals, and then some kind of like really high outliers, and then all the way in between. I think the longest time recorded was 27 minutes or something like that. So some wily lionfish out there that did not want to get caught. But yeah, there are quite a few sub-minute, around 15 to 30 seconds or something like that as well.
Okay, great. And you suggest that the smaller lionfish are harder to catch, but the bigger ones are easier to catch. Do you think it may be more of a bell curve where the smaller ones are pretty hard to catch, but also the really big ones are hard to catch because maybe they're a little smarter and have been shot at a few times?
Yeah, that's definitely a possibility where you get those lionfish who've learned to avoid divers. I didn't see that so much in these data, but I definitely know that's a possibility. And I think Steph and I had a couple of like alternating hypotheses as to whether or not you would see that exactly what you described, like a bell curve, but it was definitely more of a linear relationship where just in general, the bigger they got, the easier they were to spear. I can't remember, I think the minimum size we saw in the data sets was 4 centimeters or something, which is just like, you're not gonna spear a 4 centimeter lionfish. And so I think there's that kind of size threshold where you can't even bother trying to spear it, as opposed to the really small ones that are just right there that you can still spear, but just are harder to hit.
Yep. Okay, awesome, thank you.
Yeah.
Melanie, can you remind us what the variables in your HSM are, and were you able to conduct model selection to identify variable importance, i.e. what variables best predict suitability beyond depth?
Yes, so I have 3 variables that were the top most important variables, which started with depth, which turned out that it was kind of exactly as expected. The species is residing anywhere between super shallow 2 meters all the way down to about 50 meters of depth. Minimum salinity also came out as the second most important variable, and that showed that they can also live in brackish water habitats. And that is something that has also been backed up in research around the aquarium hobby and people trying to breed this species for aquaculture. And then the third most important variable was the current velocity in the habitats for this species, which showed that they prefer low to moderate currents in their environments. And that also made a lot of sense because the vertical distribution of this species kind of falls between cryptic and something like a brown chromis, which is moving very far off of the reef to try and catch some food. So the regal damselfish is going to stay a little bit closer to the substrate than like a brown chromis would. So they are going to be influenced by that current. So living in a habitat that has really high current velocity is going to demand a lot more energy from them on a daily basis. So trying to live somewhere where it's a little less hectic is going to be better for them.
Awesome. Thank you. All right, this is for everyone. I guess someone can chime in. We'll start with, I guess, Holden and move down the line. But when first identified as a problem, one of the goals of culling, other than the total removal, which proved to be impossible with lionfish being as deep as 1,000 feet, was the slow ecological effects and allow predators to learn to eat them. At what point do we call it the Atlantic lionfish and consider the new ecological reality?
I think that's an interesting question for, broadly for invasive species in general. And ultimately, where do we end up with lionfish? There's a lot of invasive species. Lionfish are just one of them. I think ultimately we might end up at a place where lionfish become kind of a managed fishery in some ways. We see that, for instance, in Florida, they actually manage peacock bass, which is a non-native fish, or the system does kind of balance out to some sort of biological equilibrium. I think the most important thing too, when you think about lionfish too, is to really remember that we do see negative impacts from lionfish, and that they're a part of a suite of stressors on ocean systems, particularly coral reefs. And so, I think along with overfishing, coral damage, coral disease, with a lot of other things, lionfish are one more stressor to the whole system. And so I think we really want to not, work on lionfish, but sometimes zoom out and really remember that there's a lot of other ocean conservation work that needs to be done at the same time.
Thanks, Holden. Does anyone else want to tackle that question? All right.
I agree with, I did.
Okay, go for it. Sorry.
So does Steph. So I'm going to take it really literally. And as far as actually calling it Atlantic lionfish, I would say no, never, because it sends the message that it's okay for it to be there. And we want people to continue to put energy into removing it, even if it's going to be here in the long term. And I agree with everything Holden said. So some of my other research is on, all of my other research for my dissertation is on Florida lionfish divers. And I hear from them, very different perspectives. If somebody thinks that it's not a problem anymore, they'll be less interested in removing it and putting effort into it, these spearfishers. Whereas somebody else who thinks that, for instance, the lesions that we were talking about earlier, if they think that that is just temporary and the lionfish population is going to bounce back, then they don't even talk about decreasing their effort to remove lionfish. So anyway, I also was thinking what about other invasive species in Florida? Sorry, I'm so specific to Florida. I'm from there. The Bernese python, we still call it that.
Awesome. Thank you. Go ahead, Stephanie.
One last thing to add. I think there's something really valuable in sort of recognizing that lionfish are likely here to stay, but not sort of being fully complacent with that. So just because full eradication isn't on the table doesn't mean that there aren't contexts in which management intervention is really needed or useful. And it could intersect with, as Holden said, other stressors to reefs. It could intersect with population densities of lionfish, because we know they're really patchy and sometimes are high enough to cause change and sometimes are not. And I think it's really interesting to see the direction that a lot of work in this region is going towards in terms of thinking about sustainable interventions and how to have both social and economic benefits to communities and coastal, nations from interventions, and also thinking about when and where intervention is needed in terms of spatial planning and temporal planning too. So that really encourages me that we've moved beyond that sort of, well, we have to remove every single lionfish or it's not worth it mentality towards a little bit more holistic, like integrative approach. So yeah.
Awesome. Thank you. A quick question for you, Stephanie. Do you think lionfish sex had anything to do with their movement? I mean, I couldn't really tease that out, but do you think maybe one moved more than another?
It would be a pure guess because we weren't able to determine, because you can't distinguish between males and females visually, and we weren't able, unfortunately, to recover any of the tagged fish for dissection. That was our hope. Our hope was to get vertical movement and to be able to recover some individuals that were tagged, and that didn't happen. So I have no idea. That would be really interesting. If somebody has a lot more funding to put towards one of these tracking projects, we might be able to figure it out. But yeah, no idea.
Awesome, thank you. And I guess I have just one more question for Eileen. What's the status of lionfish derbies in the Mediterranean? They've proved to be pretty successful over here.
So in Cyprus is actually the only country that has had a few derbies so far. But aside from that, there's one NGO in Northern Cyprus that's been doing weekly removals, and they've removed a lot. Anyways, we're planning to conduct our first 2 big ones over here next year, and we hope to involve you in that. So yeah, hope to see you over the pond sometime
Yeah,
soon.
that'd be great. Cool. All right, I don't see any more questions here. Give another minute for anyone to ask some questions, and we can kind of close this one up. We're just about on time too.
Thanks, Holden. Oh, a question just came in for Kaylin. If you like, Alex, you can read it out. I can read it out and you can listen to the translation. Oh, I can try.
Thanks. I'll let you do it.
Miguel, can you put on your mic and read your question to Kaylin? And Kaylin, you can listen on the interpretation.
Ok, hola. Sí, un poquito la pregunta es que si a lo largo del tiempo de la invasión que lleva el pez león, ¿ustedes consideran que la invasión del pez león ya forma parte de la cultural de la región del Caribe? ¿Ya hay cierta apropiación o de las comunidades costeras hacia la invasión del pez león?
I feel like we could all weigh in on this question because we work in all different parts of the region. And so we're thinking about, I assume here we're thinking about like in a specific community, people are already aware of lionfish, and I would love like head nodding to confirm whether this is the correct interpretation. So people are already aware of lionfish and involved. Is that sort of, do you think, Fatila?
I think the question is sort of asking like, in some cases with invasive species, they become part of the community and it becomes sort of beloved. And so
can I jump in?
Sure, Alejandro.
Alejandro, what he was referring was that due to the perseverance of the lionfish invasion and for all these years, is that the lionfish culture is becoming a culture among the fishermen. The fishermen already accepting lionfish as a part of a traditional fisheries or a cultural process. So that's what he was referring, more the acceptance from the cultural perspective and the traditional use of the resource.
I see. Gotcha. Okay, well, I'm most familiar with just a couple of parts of the region, so I can speak to those. So it looks different, especially based on context. It sounds like you have experienced or seen some acceptance in, maybe your areas of expertise. Or in Florida, there's definitely sort of like this love to hate them. They're very passionate about lionfish. It is very much a culture, and that's part of what I studied. There's a community very much based around lionfish as a theme. There are people on this call who are members of this community. And I think there's also excitement. So I've done a little bit of work in Belize as well, and there are pockets of excitement about lionfish in those places as well. I don't have specific data on this type of thing. So it would be really interesting to look at that cultural connection as well. And I also kind of wanted to open this up to everyone else because other people have experience in other specific contexts and parts of the region. Does anyone have anything to add?
I can add that there has been a change. So like for me, it's not necessarily introducing it into the traditional fishery, but I think for me, when I was working in Anguilla, at the start, the fishers were very much afraid. Lionfish, if they found them in their traps, they would just throw it away, that sort of level. And after sort of showing them the stomach contents of lionfish and that the lionfish were feeding on the fish that they wanted to eat, then they realized that lionfish were actually a problem to them. And now they do go out and hunt lionfish. And then also it's sort of become part of the traditions that are usually associated with their fisheries. So they would have like a Friday fish fry and it would be lionfish or that sort of thing. So that's just sort of a small example of it being incorporated, where especially even with their national dish, you've seen, especially if it's like a fish and rice and peas sort of thing, lionfish are being used as part of the national dish.
I could add to that actually a little bit. So in Belize, I conduct, we have conducted research on willingness to consume lionfish across the whole country. And we sort of learned that, we also looked at just generally what the traditional preferences for seafood are. And those hold pretty strong in Belize. For instance, snapper is by far and away the most popular dish as far as seafood. So sometimes it's kind of hard to break down those barriers. For the fishers themselves, it's a little bit different. I think in that particular context, for instance, it's mostly about, can I sell it? And if I can, I will. But there has to be a market. So there's two sides to this supply.
Muchas gracias a todos. Sí, efectivamente, yo considero que incluir al pez león como parte de la cultura, bueno, ya yo siento que es parte de la cultura, ¿no? Siempre y cuando no se quite el concepto, ¿no? De que es una especie invasora. Muchas de las personas ya lo integran dentro de su menú, dentro de la pesca tradicional. Hay fotos en donde se pueden ver que sacan boquinetes, pargos y hasta el pez león incluido, ¿no? Yo siento que mucha parte del éxito que se tenga de los trabajos de control de pez león es precisamente eso, ¿no? El involucramiento con las personas y que lo vean como algo que ya forma parte de la tradición o parte de la cultura del Caribe y que pues no hay que bajar la guardia, ¿no? Gracias.
Can I just say one thing? I really appreciate that perspective and the question and comments about it. And I wonder if GCFI isn't a perfect place to have further dialogue about that, including fishers from around the region, and just maybe planting the seed or like pushing the the papers over the desk for maybe the next one, that maybe there's a way to have a more formal conversation around hearing perspectives on that, because it would be really important.
Yeah, that's a really good idea, Steph. We can capitalize on the fact that we do have fishers attending our meeting, so we can look to gain in their perspectives and that sort of thing. Alex, I do see that there was a question coming in for Steph, so maybe we can just use that one as the last one. And as I'm on, I can read it. Having seen lionfish research evolve and being heavily involved with GCFI's lionfish work for over a decade, what do you see as the most important areas of research and management for lionfish and invasive species in the Gulf and Caribbean, Steph?
Oh boy, I need more coffee. I think maybe several people around this webinar could provide comment on that. But I think this conversation we're having about, beyond lionfish even, many of these marine invasions are here to stay, and understanding the context in which management is appropriate and how it can benefit people, I think is really a direction that we need participatory work in terms of understanding both the needs and the solutions that fit that. So, I presented here a tracking study, and I think that was a wonderful partnership and is valuable information, but it's how that kind of ecological data can integrate with fisheries data, with spatial planning, with understanding market forces, and evaluating the benefits and unintended consequences of those impacts, I think is really important.
Thanks, Steph. Alex, do you want to add any closing words to wrap up?
Well, thank you everyone for tuning in, and really appreciate all the talks and the great responses to the many questions we had. So if there's any other questions, feel free to reach out to the speakers individually, or you can reach out to us and we can relay the message.
Yes, thanks, Alex. And as we've said for previous sessions, these all of these were just short 3-minute snippets of a presentation, and full 10-minute presentations are available on the app. So we encourage you, if you haven't already, to please go check out those full presentations. And then also the discussions should not end here. You can continue to engage with all of our panelists and all of our attendees as well through the app. So thanks again to all our panelists and our moderator, Alex, for a really interesting discussion today. So thank you to all of you, and we're going to get started and set up with our next panel discussion, looking at exploring the habitats within the wider Caribbean. So thanks again to our lionfish folk, and I'll invite the habitats panelists to join us. Thank you.
Thank you.
Tony, would you like to take over? And as a note to Tony, we have had a cancellation. Lisa Chong will not be presenting today. So that's just a last minute update for you.
Okay, thank you for that, Fadila. All right, well, let's go ahead. That was a great panel. Thank you again to the lionfish presenters. So hello, everyone. Welcome to this session on exploring the habitats of the wider Caribbean region, with a focus on opportunities for research and sharing of best practices. My name is Tony Marshak. I'm a research associate working in support of NOAA's National Centers for Coastal Ocean Science, or NCOS. And my background is in fisheries ecology, including a focus on habitat and ecosystem science, and incorporating that information into more robust assessments and broader management approaches. During my career, I've conducted research in the Gulf of Mexico and US Caribbean, and before joining NCOS, I co-led NOAA Fisheries Habitat Science Program for 6 years. I'd like to thank the GCFI co-chairs and organizers for creating this session focused on habitat research and best practices, which is something that many of us continue to emphasize the need for and the importance of in multiple geographies. I'm coming to you from Silver Spring, Maryland, which is chillier right now than many locations from where our audience is tuning in, but it's wonderful to be here with you all today, and it's been really heartwarming to see all the exciting research that's being featured throughout this conference and the togetherness of the GCFI family, which is still coming across through this virtual platform. I'm really excited to introduce our 6 presenters and panelists to talk about their habitat-related work. Please feel welcome to put your questions in the Q&A, which we'll address after all our presenters have had a chance to give their brief overviews. And so with that, we'll jump right into our first presentation, beginning with Dr. Bob Ellis. Bob is an associate research scientist with the Florida Fish and Wildlife Conservation Commission and is interested in the forces that shape fish communities and regulate biodiversity in the ocean. And he'll be talking about his investigation into habitat use and movement patterns of juvenile Goliath grouper in South Florida estuaries. So welcome, Bob.
Good morning. Thanks, Tony. Can you hear me okay?
Yep.
Awesome. Okay, so briefly, what we've tried to do is, like the title says, we're investigating habitat use and movement patterns of juvenile Goliath grouper in a couple of different estuaries here in South Florida. We know that the Goliath grouper population here in Florida is rebounding, and it is much larger. And as a consequence of that, the juveniles have reinvaded many of the estuaries that they use for juvenile habitat. So we were really interested in how habitat use and movement might differ in different estuaries that have different levels of anthropogenic impact. You could argue that there isn't an estuary in South Florida that is not somehow impacted by humans. That can take the range from completely urbanized, where there's no remaining mangrove or seagrass habitat or oyster reefs left, it's just docks and bridge pilings, to almost completely natural state where you have most of the habitat is intact. And so what we did is we chose 2 different estuaries that kind of lie on opposite sides of that continuum, the St. Lucie River and the Fak Union Canal and River, both of which are impacted, but in much different ways. So the St. Lucie River is very heavily urbanized. You can see a picture of us fishing under a dock there. And that's entirely where we caught the fish. We caught every single fish. We tagged 30 fish in that array, acoustic array, and they were all under docks and rivers, docks and bridge pilings. In contrast, the Fak Union area is part of the 10,000 Islands. It's extensively fringing mangrove habitat that's largely intact. And the anthropogenic impact comes from changes to the water flow patterns. So we caught all of those fish. We tagged 20 fish in that array, and those were all caught underneath overhanging mangroves, places where the river was undercut and the mangroves come right up to the river edge. And so basically the fish are using a very similar physical habitat, but that is vertical structure, but it differed pretty dramatically in terms of what made that vertical structure. So for the purposes of this talk, I really wanted to focus on some of the broader scale patterns that we're finding. We expected that changes in water flow and salinity would be the main driver of fish movement, particularly in the St. Lucie River. That river and the location where we tag the fish goes completely fresh when water gets released from Lake Okeechobee and is channelized down the river. And so we expected to find fish moving in response to that. And in fact, what we found was that fish were largely, they pretty much didn't move when that happened, but we found a really interesting response to a hurricane. Hurricane Irma came right through and dumped a ton of water on the watershed, and we did see fish moving in response to that. The other thing that we found that was very interesting was that temperature seemed to be the main factor regulating movement in these fish. And in the 2 arrays, it was completely different. And so that's what these graphs are showing you here. Basically, you have temperature increasing on the x-axis and then the variance in location on the y-axis. And you can see that on the left-hand side is the St. Lucie River. As the temperature increased, we actually saw fish moving using more of the river array, whereas in the FAC Union temperature increase, we saw fish moving into the river and using a smaller part of the array. So we're really interested to dig more into this data and find out, what exactly is driving these patterns, and how this could be tied back into some of the water use management that we have here in South Florida. And I'll leave it right there.
All right, well, thank you, Bob, for that great overview. It's really informative, very interesting work on Goliath grouper habitat patterns. Next, we have Dr. Antonella Rivera, who's a PI at the Coral Reef Alliance, and she works on generating research to support science-based decision-making in fisheries and water quality of the Mesoamerican Reef region. And she'll be talking about a case study in Half Moon Bay, Honduras, that focuses on the impact of sanitation best management practices and human and coral reef health. So welcome, Antonella, the mic is yours.
Thank you. Well, as Tony mentioned, I'm here to talk to you about how sanitation best management practices are impacting both human and coral reef health. And this is a collaboration between the Coral Reef Alliance, the Bay Islands Conservation Association, Healthy Reefs Initiative, and Polos Water Board. So as you all probably know, the Mesoamerican Reef region is the second largest barrier reef in the world, and it's critically endangered. One of the main threats that it's facing is an increase in macroalgae, which has nearly doubled in the past decade. And one of the main causes for this doubling of fleshy macroalgae is usually poor water quality in the region. But one place which has successfully established sanitation best management practices and implemented them is Half Moon Bay in Roatan, which is part of the Bay Islands of Honduras. So what we did to collect information on this case study was first to systemize local ecological knowledge in the area. We also used water quality data, which was collected since 2013 by BICA. And we analyzed coral reef data, specifically coral disease from 2005 to 2018, which was collected by the Healthy Reefs Initiative. And what we found was that the process of established sanitation best management practices was quite long, and it had many ups and downs. It started in 2005 with a dissatisfied community. And then by 2006, they established from the bottom up a water board, which took quite a long time to gain the trust of the community and make sure that they could garner community support. But all this work paid off because by 2012, they were able to successfully establish a sewage treatment plant. And currently, in 2021, 99.65% of accessible homes and businesses are connected to the sewage treatment plant. So what was the use of all these sanitation best management practices? Did they actually have an effect? And as you can see on the graph there, we have average enterococci concentration per 100 milliliters, so basically bacteria in the water, per year. So each bar represents the year. And what we started seeing is that before the implementation of the sewage treatment plant, bacteria in the water was quite high. But just a year afterwards, we already see a very sharp decrease in these bacteria, which is correlated to the number of connections to the sewage treatment plant. And by 2017, they are already meeting the Cartagena Convention limit. And actually, they currently hold a blue flag bleach award because of the way that they've been handling the water quality in the area. But not only that, when we looked at the data for coral reef health as well, we saw that in 2010, before the establishment of the sewage treatment plant, bacteria was quite high in the area. And that also meant that disease for coral reefs was quite high. It was around 15 to 25%. And after the implementation of the sewage treatment plant, so when we looked at it at 2016, disease had gone down to 0, and it has remained at 0 2 years afterwards. So basically what we saw in Half Moon Bay is that sewage treatment plants work, and they're a cornerstone for sanitation best management practices. However, they're not the only factor that we need to consider. These types of practices must be locally tailored, adaptive, and require trust, as well as the involvement of all stakeholders. So if I can leave you with just one takeaway here today is that what we have seen in Half Moon Bay is that sanitation best management practices work, and they're important for both human and coral reef health. Thank you.
All right. Thank you, Antonella. That was an excellent overview, and it's quite encouraging to hear about how wastewater treatment and stakeholder engagement and emerging best management practices are leading to positive results. Our next presentation is from Dr. Kelly Kingen, who is an assistant professor at the University of Trinidad and Tobago. with broad research interests in uncovering fish movement patterns via telemetry techniques and mapping marine habitats at multiple scales using a variety of remote sensing techniques and GIS tools. Kelly's work is about the fish and invertebrate community associated with invasive seagrass and St. Eustatius. So please welcome Kelly.
Thank you, Tony, for the introduction. And thank you everyone for coming. I'm excited to give this presentation this morning. So this work was a collaboration between STNAPA and St. Eustatius, several citizen scientists, a volunteer who helped analyze the data, and myself. So I actually haven't been to St. Eustatius, so I kind of organized this virtual project online, and I'm eternally grateful for my co-authors here for all their assistance. They collected a huge amount of data in a very short amount of time. So my team surveyed seagrass beds at various depths on the leeward coast of St. Eustatius using belt transects and quadrant photos to quantify the fishes, macroinvertebrates, and algae found within the transects and the quadrats, and all the well, I shouldn't say all the seagrass, but mostly targeting the alien seagrass species Halophila stipulaceae. What we found, the most dominant fishes were the rosy razorfish, in which we encountered over 1,300. The dominant macroinvertebrates were cirrus snails, of which we found abundances or densities up to 250 per meter, so quite prominent. And the dominant algae were filamentous red algae species, which accounted for about 30% coverage across 3 different species. And 2 of those species can be seen in the upper left photo here. And in the middle is the rosy razorfish, and right, upper right, the cirrus snails. We covered a range of depths from 8.5 to 22 meters. And then we compared the fish diversity we found and fish abundances to depth, but we found no relationship with depth in these instances. We're still analyzing to see if there may be community shifts with depth and if there are relationships with depth and algae or the macroinvertebrates. As far as the native seagrasses go, they were quite rare. I shouldn't say rare, but only really seen in the shallower depths. And the 2 species we saw were manatee and shoalgrass. And they were always intermixed with the H. stipulaceae and at very, very low coverages, less than 2%. We saw no turtle grass at all. In total, we encountered 189 species, but 153 of these were directly above or on the H. stipulaceae meadows. We also saw several ecologically and economically important species like queen conch, juvenile snappers, big barracuda, like you can see sneaking up behind me, long-spined sea urchins, surgeon fishes, parrotfishes, things like that. So the introduced species seems to be providing important habitat for a diverse community and into deeper depths and kind of more polluted areas, like around the harbor area in near Ranchestad, than native seagrasses. And with that, hopefully I've made my 3 minutes. Thank you guys.
Thank you, Kelly. That's great, very informative, super topical and very interesting work, especially as Halophila continues to invade the Caribbean. Our next presenter is Dr. Francesca Pella, an environmental data analyst with Proges Consulting, who's joining us from the United Kingdom. And she'll be giving us an overview of her presentation, The Challenge to be Sustainable, Systemic, and Synergic, Lessons Learned from the Caribbean DSS. Take it away, Francesca.
Thank you. Good morning or good afternoon, everyone. My name is Francesca Pella. I'm freelance. In the last years, I've been involved in different ecosystem-based management projects. First of all, I would like to say that I'm very happy to participate to the conference, to be part of this panel, and also my congratulations to the organizers for the well-planned conference. The co-authors of this presentation, this work, are Dr. Nina Lisenko and Pedro Matteo from the Vice Ministry of Coastal and Marine Resources of Dominican Republic, and Marco Falcetta and Eduardo Scepi from Project Company in Italy. The main EBM DSS experience described in our presentation was applied in Dominican Republic. Was a pilot project that was implemented by United Nations Environment and under the coordination of the Ministry of the Environment and Natural Resources in Dominican Republic. The other mentioned project, still ongoing, is focused more in the Mediterranean area and is called MED for EBM. What I would really like in these 3 minutes is in the context, of course, of the ecosystem-based management, is asking to the participants to share their experiences, in particular focusing on 3 aspects. The first is the data, storage and management data. The second would be the stakeholders' participation, and the third, the synergies with the other projects. So all we know that the collection of the data can be challenging and possibly related to the data availability, accessibility, and format. What is or what was your tool to store and consult the data? This is my first question to the participant. The second is about the stakeholders. And we all know that in particular in the EBM process, the stakeholders' participation is crucial, is essential. It is also a quid pro quo relations. So the stakeholders are there to share the information and the data, but at the same time, they follow the process of the EBM. So they gain awareness and knowledge and ownership of the EBM process and tools. So my question in this case, in your opinion, what is the main challenge in this aspect, in this perspective? And the third bullet is about the synergies and the sustainability. So there are several examples of synergies that can improve and or guarantee the sustainability of the EBM projects. For example, nowadays, the Vice Ministry of the Coastal and Marine Resources of Dominican Republic is still disseminating the tools and the concepts and the methodology of EBM applied in our process, in our project, sorry. But at the same time is contacting other projects to promote synergies and to maintain exactly the sustainability of this approach. So my question in this case is, in your experience, what is the role of communication that is playing in this respect? And I thank you for your attention, and I'm available, of course, to answer to your questions. Thank you.
All right, well, thank you, Francesca. It was a great overview of engaging with stakeholders, some good questions to think about for further discussion, and great to see applications of stakeholder information into ecosystem-based management. Next, let's please welcome Iris George, who's an undergrad student at the University of Alberta. And Iris is finishing up her degree in ecology, says she's in love with all things marine, and is very excited to begin sharing her research. Iris will be providing an overview about her work on determining drivers of detectability for reef species between marine survey techniques. So welcome, Iris.
Awesome, thank you so much. So yes, I am Iris. I'm an undergrad student from the University of Alberta, and I'm just going to give a very brief overview of the work I've been doing with the Change Lab, examining the effects of species traits and habitat quality on differences in fish detectability between visual survey techniques and the implications that these effects might have for coral reef monitoring design. So the important role of the fish community on coral reefs is really well understood, both ecologically and economically. And regularly and accurately monitoring reef fish is very important because of this, but there are several different visual surveying techniques that are currently used to accomplish this goal. So we examined and compared 3 visual surveying techniques in this study using data from 2014 to 2015 from the Florida Keys region. But today I am just going to be talking about 2 of those techniques. So the first we explored were stationary visual censuses, or SVCs, which are shown to the left of the slide in that diagram. And these are performed by a stationary diver. And the second method were belt transect surveys, which are showed to the image on the right of the slide. And these are performed by a moving diver swimming in a straight line. So these techniques do differ in their efficiency and their detail of the reef coverage. And we really wanted to explore how these impacted their detection of fish. But instead of seeing how they varied for specific species of fish, we explored their differences in relation to physiological and behavioral traits of the fish species and also habitat traits of the survey sites themselves. So we compared the fish densities captured by those SPC surveys, which are the least detailed but the most time efficient method, to the belt transect surveys, which do provide a more detailed reef coverage. We performed linear mixed effects models and subsequent model selection on the density differences between SVC and belt transect surveys in relation to those fish and habitat traits. And we found that belt transect surveys were consistently capturing higher fish densities of all trait groups we looked at compared to SVC surveys. However, the magnitude of those differences did vary with both fish and habitat traits. So for example, we saw SVC surveys consistently capturing lower densities of camouflaged and small-bodied fish compared to larger and more colorful or silvering individuals. So very overall, despite the efficient nature of the SVC surveys, we saw that they were unable to capture a balanced composition of the reef fish community. And because of that, we recommend that their data be used to monitor larger non-cryptic species, which do include many groups of commercial importance. So these are still a really useful technique. And we recommend that despite the increased time and training required to conduct the belt transect surveys, these should probably be used to monitor the community as a whole. So our results highlight that the most appropriate survey method to use is going to vary with the management goal of the project, as well as the specific traits of the fish species and habitat types being targeted by the surveys. So all of these must be kept in mind by managers when choosing a survey method in order to ensure that whatever method does end up being chosen is leading to the most accurate results, which is what's going to ensure the ecological and economic health of the reef fish community into the future. So that was very brief, I know, but yeah, that's in a nutshell. There's a lot more detail in the presentation and also in our manuscript coming up. But yeah, thank you so much to GCFI for the opportunity to share this. It's been great.
Excellent, thank you, Iris. That's a great interview. I really enjoyed watching the recorded presentation. That was a great study on the effects of habitat and body morphology variables for conducting effective surveys. And continuing with our final presenter, we have Lizzie McNamee, a researcher and diving and boating safety officer from Florida Atlantic University. Lizzie was previously at Florida International University. She'll be talking to us about food web modeling to assess interactions between artificial and natural reefs. Welcome, Lizzie.
Thank you. Can you hear me?
Yes, I can.
Awesome. So as Tony said, my name is Lizzie McNamee. I'm with Florida Atlantic University, and this project was for my graduate work in conjunction with a bigger research project with Florida International University. Previous studies show that artificial reefs attract an abundance of reef-associated fish. They're deployed worldwide for both diving and fishing purposes, but there's a significant lack of comprehensive information related to regulation, long-term management goals, and environmental assessment, including how they impact the underwater communities that they're deployed in. We really wanted to investigate how artificial reefs may interact with the surrounding communities, knowing that these structures just don't exist in a bubble. We looked at the artificial reef in the Florida Keys called Aquarius Reef Base, pictured right there in the bottom in the middle, and a number of natural reef sites radiating around it. Fish survey data was input into EcoPath with a number of morphometric components and food web information. Looking at the figure on the left, we saw more fish in total, more pelagic fish and bigger schools of fish were observed on the artificial reef. It's really important to notice the 2 orders of magnitude change in scale for Aquarius Reef Base there. Through modeling, we show that there is just not enough food on Aquarius and that the predatory fish that reside there must forage off of the artificial reef and onto the nearby natural reef. Next, we mapped the seascape of the surrounding natural reef seen there in the middle on the top, identifying the major reef types and introduced the artificial reef food web to equivalent areas of biomass from each seascape. None of the habitat types could provide enough food or enough variety of food to satisfy the population at the artificial reef. Lastly, we introduced the habitats in the proportions they are available. By looking at ecotrophic efficiencies, which can represent overexploitation, this is explained a little bit more in depth in my pre-recorded talk. We were able to show that the population that shows residency on Aquarius requires over 3 and upwards of 6 kilometers of surrounding seascape to access enough quantity and variety of prey. These findings were supported by acoustic tracking of a couple species of predatory fish, which showed residency on Aquarius with consistent utilization of the closest natural reef sites. This is the first study that we know of that uses this methodology to investigate the spatial subsidy of artificial reefs by natural reefs. And the tool will allow for easy inclusion of spatial and temporal mapping and extractive practices such as fisheries. Future studies on other artificial reefs really need to be done. These would include variables such as the footprint size of the artificial reef, vertical relief, and the distance from the natural reef. This is really important for ecological and management implications for future deployments of artificial reef, especially here in the Florida Keys National Marine Sanctuary, where we're the only national marine sanctuary that allows for the purposeful deployment of artificial reefs. And these artificial reefs should be purpose-driven as to balance ecological and economical drivers. Thank you, and I look forward to your questions.
All right, thank you so much, Lizzie. That was an excellent overview and a really nice comprehensive study, very interesting work about natural and artificial habitats in the Florida Keys. So that brings us to the end of our speakers' short overviews of their work, and we can go ahead with our Q&A session. Again, we invite our attendees to please enter any questions they may have into the Q&A option or the chat. And we also welcome our panelists to ask any questions they may have for each other too. We're already getting some questions that are popping up, so we'll go ahead and move right into those. First question is for Bob Ellis from Mark Peterson, and question of how did the frontal pressure of storm influence movement? It's been shown that fishes can detect these fronts as they normally move.
Yeah, that's a really great point. So we actually saw that immediately before the storm passed that the fish were utilizing habitats furthest up the river in the St. Lucie than we saw pretty much during any other time period during the study. You know, with 10 minutes, you can't really get super down into the details, and I wish I could have included more of the individual fish plots. But we absolutely did see fish concentrating in the river itself before moving out of the river as the salinity dropped. And again, that was, we only have that one storm that went across. We had maybe a dozen fish tagged at that point in late 2017. So unfortunately it's unreplicated, but we did definitely see storm related movements. Fish moving upriver and then downriver as after the storm passed.
Okay, great. Thank you, Bob. We have some questions that have come in from Michelle Scherer as well. The first is to Kelly. Question of what was the density of juvenile conch in the Halophila seagrass compared to the density of queen conch in natural seagrass habitats?
Hi, Michelle. Thanks for the question. So our density of conchs was quite low. I think we saw 6 in total across our 24 30-meter transects. So not a whole lot, but they were there. Sorry, there was another part to that question, wasn't there? Oh, did we compare it to native habitats? Unfortunately, there doesn't seem to be any like monospecific fields of any of the native species in St. Eustatius, so we couldn't really do a comparative survey. The native species are now just intermixed with Halophila, and they're in low density, so it's really the only habitat for the conchs there at this point, as far as seagrass habitat goes anyways.
All right, thank you, Kelly. Also from Michelle to Lizzie, how much of that predator biomass on artificial reefs were really residents versus those that were aggregating for reproduction or spawning?
Absolutely, great question. We actually looked at this as part of the greater multifaceted project that my research was part of. We tagged a whole bunch of barracuda and schoolmaster snapper and tracked them along the natural reef and artificial reef for extended amounts of time. And we found that all of the tagged fish showed residency on Aquarius reef base or artificial reef, and showed foraging on the nearby natural reef, mostly to the natural reef sites closest to Aquarius reef base. Our snappers showed clear diurnal migration away from the artificial reef at nighttime to forage and back on during the day. And the barracuda were more variable in their movements, but they did show high water residency during the day and then deeper water foraging at nighttime away from the artificial reef. Now, this is only one artificial reef, and it's a very specific one that is in a research only zone because NASA and the Navy use it. So no one's really allowed there. Technically, we see people there, unfortunately, illegally. But this kind of research would be very important, let's say on the Jupiter wrecks, where we know for a fact our Goliath groupers will migrate on and off depending on the season. The really cool thing about EcoPath is that we can put in those spatial and temporal factors as well as fishing factors quite easily once you get the structure of the model set up.
All right, great. Thank you, Lizzie. So yes, we continue to invite our participants to please put in any more questions into the Q&A, but I certainly have plenty of questions for our panelists as well, and I really enjoyed watching everybody's recorded presentations. So I think I'll turn to Bob with a question. So in addition to the impacts that we're seeing with mangrove loss and also concerns about water properties in Florida, have you been able to detect any effects of red tides or perhaps cyanobacterial blooms in freshwater systems on Goliath grouper during their early life histories?
So not specifically during this project. Cyanobacteria blooms are a concern, especially in the St. Lucie system. But there has not been, or there hasn't been one while we've had fish being tracked in that system. We do see the red tide impacts that we have seen on Goliath grouper are pretty variable and interestingly seem to be pretty well distributed amongst all size classes. So this past summer, we had a pretty significant red tide bloom right here where I'm located in St. Petersburg. The red tide actually came into Tampa Bay, and the hypothesis is basically that it trapped a lot of fish from being able to exit the bay. And we found a lot of large adults that died from that event. Presumably they were using some of those bridge pilings and then like I said, got trapped as the red tide moved in. There was nowhere for them to go. And so then they succumbed to it. We've seen that in 2017, we had a large red tide in the Gulf as well. And we saw the same thing, that it's pretty well distributed across all sizes of Goliath. So if there's adults that happen to be using inshore habitats during that time, which we're finding increasingly is true in the Gulf, that they're highly susceptible. Interestingly, we rarely find very small ones, and we've been curious about this as to whether or not that's a sampling effect. Perhaps we just miss them. Whenever a big one washes up on a beach, we get numerous phone calls about it, but a smaller one may just wash out with the tide and not be noticed. Or perhaps being closer up in these sort of upriver habitats may actually confer some protection. We know that snook, for example, will move into rivers and are more highly resilient to a red tide than some of the other species that can't handle fresher water like that. Red tide does very poorly in freshwater. And so you rarely see it moving into upriver habitats. And so these rivers kind of serve as a pseudo refuge for some of those fish that can move into more brackish water.
Okay, thank you, Bob. I appreciate that overview. It's very interesting. We've had another question come in for Antonella, which is, what was the approximate cost of investment and yearly maintenance of this water treatment system?
Yeah, thanks for the question. It varies a lot. So as I think I was mentioning in a question answered directly before, this has been a very collaborative process, and it's not just funds from the water board, which is how it usually works, but we've been joining funds from philanthropic organizations, NGOs, and the local government. And usually the way it would work is that for the first years up to 2020, the water board had to hire someone else to do the maintenance for the plant. But they did take the time to train new personnel and getting the equipment necessary. And now the water board does all the maintenance itself. So that reduced a lot of the cost. But I would have to double check on the exact amounts, because as I said, it does vary a lot. And because it comes from different sources, it's hard to really pinpoint the exact amount.
Okay. Thank you, Antonella. I suppose I have a question for you as well, Antonella. We have you here. I was really appreciated some of the metrics and the timelines that you were showing in your presentation and slide. I was curious if you observed any direct changes in terms of, say, epiphytes on seagrasses or algal cover in coral reef habitats over the past decade since the construction of that treatment site.
We haven't looked at epiphytes yet, but algal cover, we have seen an increase in coral cover. And obviously a decrease in fleshy macroalgae cover. However, it hasn't been equal in all the 3 sites which we usually survey. So that's why we just presented the data for the coral disease, which is the clearest answer we have, but we have seen these important changes. And what's interesting is that it's in these sites of the island. This is kind of the western side of Roatan. And when we look at other sites in those areas, fleshy macroalgae cover has increased instead.
Okay. Thanks. Thank
Can
you.
I ask a question as well, since I'm already
Of course, certainly.
For Francesca, I really enjoyed your presentation, Francesca, and I was wondering, because you systemized all this like knowledge from the stakeholders, what was the process afterward to return that knowledge to them?
Yeah. Thank you for the question. To manage the data, to collect the data better, we used we can say a double methodology. The first was the help of stakeholders. So we organized the workshops and to ask them first, like in my presentation, I summarized in my presentation, to create a sort of simple system where they had the opportunity to describe all the aspects, from the socioeconomic aspects to the ecosystem aspects of the target areas. Then for each of them, they are invited again to list all the indicators that the institution are already using for the daily monitoring, and so linked with the data. The stakeholders had the data in their hands. They could put available to the project, or of course they needed a sort of formalization, an agreement with the institution to use this, to provide the data. And so we used also a tool that is called Integrated Spatial Planning, as a software that is able to, of course, after a training and a work done with the stakeholders, with the interdisciplinary group, is able to store the data and put a sort of systematization of the information in a way that you can imagine after all this work, you have a software where is visible not only the data of the single aspect, that overlapped with the data from different aspects. Why is this? Because this tool is useful also for management purposes. When you are after the data collection, you have to show, and then I'm answering to your question, you have to, first of all, show the outcome to the stakeholders. because they have to propose the action plans based as much as possible on the quantitative information that they provided, so they can see that their effort is used for a practical purposes. And at the same time, also the agreement with the institutions, between the institution and the Minister of the Environment, that is the main coordinator and user and manager of this software, make them available to see the data. Then there are different layers, we can say different levels. So the ministry is the coordinator and the manager of the data, is responsible to update the information, to keep in contact, like I said, to continue to invest in synergies to make this system sustainable and long-term efficient. And then at the same time, the institution that provide the data have the possibility to see the data in this software. So they can use the software for their management purposes. And so it's practically, they are working on create like one big store that is dynamic and adaptive. Every time that a different project that is not only the ecosystem-based management. For example, now they are in contact with a new GF project in a different province respected to the pilot area, and focus on the watershed management. Sorry. So these different parts of the software, we can say, they are updating and adapting to respond, to enlarge the umbrella, the influence of this, yes, and to increase the not only the quantity of data, but also to answer to the different questions that the stakeholders could have.
All right, excellent. Thank you, Francesca. Appreciate that. And going to go to Lizzie at the moment. We've had a question come in from Joanna Pitt. Quite a few questions, so I'll try and go through this quite slowly. Do you think the structure of the Aquarius site, which appears relatively large, but relatively simple, affected the fish community there? And might there have been more fodder fish associated with a more complex structure with a range of holes or refuge sizes?
That's a super awesome and really important question. Aquarius reef base is surprisingly small compared to a lot of artificial reefs out there. We have a 84 square meter footprint for the artificial reef compared to, let's say, the Vandenberg, which was deployed off of Key West, which is 527 feet long, a massive naval ship. So our suggestion would be to do this type of research on varieties of different sizes of artificial reefs, from small and close to natural reefs, like Aquarius is. It was put in specifically for studying. It's an underwater habitat where researchers can actually live underwater, all the way up to very large, maybe farther away from natural reefs, to see really what the footprint and that kind of buffer zone between the artificial reef and the natural reef needs to be. This was only one beginning research study looking at the effect of artificial reefs, taking into consideration the natural community. So we really suggest the same type of study on other reefs. Aquarius reef base, as you suggested, is very unique. It's a small footprint, but it's quite tall and has a lot of understructure, which makes it really, really great for those larger barracuda and snappers, kind of protecting themselves from that current, the Gulf Stream, which can rip by, to conserve energy so they don't have to swim around and forage a lot. Having maybe lower vertical relief with more small holes for our, as you say, fodder fish is a great idea if we want the purpose of this artificial reef to be for reef rehabilitation, or for divers and snorkelers to really appreciate some of our smaller, more beautiful reef fish. And really that's kind of where that purpose-driven analysis of the artificial reef should go into. Do we want it for big commercially viable species? Great, put it far away from natural reef and make it big. Do we want it for small reef fish, for divers, snorkelers to enjoy? Make it a little bit smaller, give it those nooks and crannies. Maybe we won't have as much impact on the surrounding natural reef in those cases.
Okay, great. And I think just to follow up from Joanna as well, I think you got into her third question about each artificial reef site being evaluated individually, but any guidelines that you would suggest for future artificial structures that are proposed for reefing?
Really, really important to ask the question of what is the purpose of this artificial reef? And we're always going to have to balance the give and take of, can we knowingly impact the natural reef, and is it okay? So if we're going to put these artificial reefs close to the natural reef, knowingly impacting the natural reef, is it viable? So using Aquarius Reef Base as a good example, it was put specifically by the natural reef to be able to have long-term ecological studies of reef and reef fishes. And in those cases, we can consider it to be an appropriate use of those pros and cons of putting it near natural reefs. But if it's not for studying or anything like that, if it's for, let's say, bringing in big commercially viable species, putting it far away over 3 to 5 kilometers, because we're finding that this impact is quite localized. That buffer zone, or that ecological halo, as one researcher put it, is quite small. So putting it farther away would be great. I'm sad that Lisa Chong isn't here, because her study also supports this research, saying that the farther from the natural reefs these artificial reefs are, they benefit the most biologically for commercial fishing. And I was just like, that's awesome. We support each other's studies. So it's really cool to see that.
Excellent. Thank you, Lizzie. And yes, certainly, if you do get a chance to see Lisa Chong's presentation, please take a look at it. It was really good socio-ecological model that does get at some of those questions and really feeds in a lot of those variables. Also thinking about even how size, structure, and those kinds of factors also influence those overall dynamics between artificial and natural reefs. So we've had quite a few questions popping up in the chat, which is great. First one is to Iris from Alejandro Acosta. Question of, how do your results compare to previous studies comparing visual census methodologies, and what would you recommend to enhance existing survey protocols?
Awesome. Okay, thank you for the question. So yeah, firstly, in comparison to previous studies, we have obviously looked at those, and I'm not going to be able to recall exact names of authors and things like that, so I apologize. But in general, there is actually some variation in comparison, especially the SVCs to those belt transect surveys. There is variation in what past studies have found in comparing the densities captured by those two. So I have seen kind of all across the board, I've seen ones that have found SVC surveys capturing higher densities as opposed to belt transect surveys. So like the opposite of what we were seeing overall. And I've also seen several that find comparable densities between the two. So they find quite similar performance. And I don't have a great explanation for the variation there. I think like ours, we've broken it down into those species and habitat traits. So we have it kind of in smaller pieces, I guess, is a possibility for why we're seeing something different there, as opposed to a lot of other studies are just kind of in general looking at kind of total species density differences. So yeah, there is some variation there. One thing that's consistent in our results to previous studies is there have been quite a few past studies that have seen that SVC surveys do tend to underestimate the densities of those small cryptic species. So we're continuing to see that as kind of the main trend in our SVC bias, is that they aren't picking up the small bodied and cryptic species very well. So that's remained similar. But yeah, there's definitely some variation. And yeah, so for the second question, in terms of what I would recommend to enhance existing survey protocols, I think my main takeaway from doing this has just, and I haven't had the opportunity to actually perform these surveys myself, so I definitely am not I'm not able to give like a fully comprehensive answer, I don't feel like, but I think just really focusing in on your question and trying to tailor whatever method you're using to what you are actually trying to find out, instead of just necessarily taking a survey that that has been used a lot in the past, that has like really defined protocols, and applying that to your study or your survey, trying to think about what you're actually trying to get out of your project. And if there are specific species you're trying to target, then that's a super important thing to recognize and to think about, you know, their behaviors, where you're going to find them on the reef. And yeah, if they're going to be kind of tucked away somewhere, if they're smaller, more benthic, then an SBC survey might not be the way to go for you. You might want to try something that is getting that more detailed coverage. But yeah, if it's a more commercial species that's demersal, kind of hanging out in the water column, that's easier to spot, then you might be okay just sticking with SBC. But yeah, I guess just considering the implications of the method that you do choose and making sure that you are picking one that is going to give you the most accurate results. So kind of a long-winded, but.
That's okay. Thank you, Iris. That's great. Very, very
May
great.
I ask a follow-up?
For sure.
Certainly.
What are your feelings, or how do you think about if, let's say you're doing surveys on a variety of different habitats, do you think that you could change what SVC you use for the one that's most useful in that habitat type, or would it be kind of less consistent to get the information?
That's a really good question. Yeah, if it's all part of the same study. Because I could see that would definitely like complicate any type of analyses you were wanting to perform on that, definitely. I guess I don't know. I think maybe it would depend on like how drastic the differences between habitats are, perhaps. I think overall in a full study, consistency is going to be the easiest way to go. But yeah, like we are seeing that you might, depending on habitat types, you might be seeing more bias on certain reefs than others. So yeah, that's a really good question. That might be something that we can, or someone can look into. Yeah.
Great. Thank you, Iris. I move to a question that's for Kelly, actually from Bob Ellis. Any idea how the results at the invasive seagrass beds compare to beds dominated by the native species?
Hey, Bob. Thanks for the question. So good to see you. And excellent study on the Goliath group are really interesting. As far as looking, like I said before, there's not any native seagrasses in St. Eustatius to kind of compare this to, at least in these monospecific beds. But looking at other studies that have compared the two species, it's highly variable, depending on one, the methodology, and two, the area. Seems to kind of lead to different species dominating one habitat or the other. So some species have found higher diversity in Thalassia or turtle grass, whereas others have actually found the opposite and actually found higher diversity and higher abundances in this invasive seagrass than the native seagrasses. So it's real interesting. There, as far as I can tell, there doesn't seem to be any patterns that makes sense at this point. It just seems to be kind of island by island what kind of happens. And uniquely in St. Eustatius, these razorfish are all over the place, and you really don't see them in most seagrasses I've been to. So it's kind of a unique community just within this one little tiny islet. So yeah, I'm not sure how that's going to pan out as more of these studies occur. There's also a study on looking at the species as kind of a colonizer, and that with succession, we might actually see communities shift back to more native species, because they tend to like areas that are polluted, that have high nutrients, and they might be able to kind of take those nutrients out of the system and make it a more favorable environment for the native species to actually come back. So I think there's study to come in like the long-term effects and how the communities are going to shift over time.
Great. Along those lines, we had a question come in from Ron Hill asking if there were natural or native seagrass species in the past in St. Eustatius.
Yeah, there were in the past. I'm not really sure what happened to them, but apparently turtle grass has been gone for quite some time. I think before the invasion of Alapilla. So I don't know if it's because of nutrient pollution or if it's because of tourists coming through. I really have no idea what happened to it. My co-authors could probably answer that question a little better that are based there. But I assume before Alapilla, the Syringodium, the manatee grass and the shoal grass were much more predominant, but they've been kind of out-competed.
Sure. I just had a question as well on the subject. I noticed in your recorded presentation in your talk, you talked about doing more visual kind of surveys and photo analysis in the halophila beds. I was curious if you're also looking at other kinds of techniques like drop traps or perhaps small trawls or other types of techniques to get at some of the more cryptic species or some of the other components of the community, mesograzers, things like that.
Well, I had to use the resources that were available at SNAPA. So they had transects and quadrats on hand. And basically for my citizen scientists are well-known underwater photographers. Well, at least two of them are. So photography just was a really good fit for this survey. I think it would be interesting to do some trawls. I haven't seen any studies where they've actually done trawls on this species. On these beds. So I'd be real curious what they find in the trawls because you get a lot more fishes in the trawls than you're going to see in the seagrasses or get in traps. Like most of the traps surveys I've seen gets very little diversity of fishes in those traps. So I think that work is needed and would be really interesting to compare if we can get some trawls or some other gear types there to try it out. But I'd be keen.
Great. All right. Thank you. We've had a couple other questions come in for Iris. One from Bob Ellis. Did you look at the influence of total fish abundance on the efficacy of the SVC method on capturing cryptic fishes? Another question, getting back to the cryptic fishes question. Having recently done SVC type surveys at sites with low total abundance due to red tide, we actually found a higher abundance and diversity of small demersal fishes. And we're concerned that this may be an effect of the survey type rather than representing the true abundance of those fish.
So that's really, really interesting. I haven't considered that, but I might have to now because that sounds like a really, really good point. But yes, so I guess the only kind of the only suggestion I might have for what you've personally experienced doing those surveys is I have seen SVCs can be biased if any of those species are schooling or something like that, like passing through the survey area in larger groups, even if they are smaller individuals, then that might be a possibility for why you're seeing such high abundances of those. But yeah, it's a great point, and that's really an interesting observation. So yeah.
Okay, great. And Ron Hill also has a question for you, Iris. Stationary surveys have been known to be less effective at picking up cryptic species, but have been better at actually defining a known area to survey compared to belt transects. And was well-defined area a factor in your analysis?
So again, that wasn't something that we had in those models. So our sites were just based, like our survey sites are defined sites on the reef. And we were just kind of like the surveys that were performed for that site were applied to the reef as a whole in that area. So yes, that wasn't something we looked at. I'm not sure if we have the data necessary to include that as a factor, but that also may be something I go and check out now. But yeah, and that's a good point. I was really, I'm happy to hear everybody else's experience doing these, because I know they are, like, I mean, SVCs are by far the most common visual surveying method. So it's great to hear everybody's experience with them. Yeah.
Excellent. Great. Thank you, Iris. I have a question for Francesca, actually. I'm really appreciating the work that you're doing and engaging with stakeholders and also trying to apply that information to topics like ecosystem-based management. And this is something that I guess I've struggled with too, and the best way to engage with stakeholders who are often, focused on a particular species or sector or particular environmental factor. And it can be kind of challenging to communicate ecosystem-based management from that perspective. What did you find was most effective for engaging with stakeholders and informing them about how their information could be applied toward EBM?
In my experience, it's always been in different countries a long process. At the beginning, we invited all the stakeholders again from different institutions, but also from the from the university professor to the simple, we can say, to the normal professional, all sort of people that can share their experience, whatever it was the origin of work or professional dimension. In the first workshops, we realized that it was very helpful. For example, let them speak about their experience, their work, the area from their point of view. And it was useful for us to understand better the system and to start to create this graphical representation that I call diagram in the presentation. And at the same time for them, because they soon realize, this is the first challenge, that often the same words means different things from different stakeholders. So when, for example, we mention a certain aspect of the area, they were invited to define, to find a shared definition. So first, this is a very, we can say, basic and possibly banal example, but it was very effective because then they share they find they built the definition of a certain aspect. And then systematically, we went through all the aspects to describe. So this was the first the process to build a common vocabulary. It was definitely one of the steps. I don't know if and then of course the thing that I mentioned before, the quid pro quo. So they share their information and at the same time they built a sort of better knowledge of the EBM because they start to realize during this process that their sector is linked with other sectors. They knew already before, of course, but they were better aware. We were better aware, of course, and also the process through the way that they were invited to list the indicators and to list the data, to provide the data, invited them to become more pragmatical. So they can see that the relation was not a sort of relation in spread in the universe, but they had a definitely very complicated sometimes and not always direct correlation between one aspect and the others. So this was another series of discussions that brought them to understand better we understood better the ecosystem-based management in this way. I don't know if you share the similar experience or what is your point of view, I mean, in this respect.
Certainly, and I'd be happy to follow up with you. I've had a couple questions come in as well. I think one for Antonella. With the shift of everyone to the sewage treatment system, is there a plan to extend monitoring to track future changes in the system over time? And also, is there a plan to share the results of the project regionally to other communities?
Great question. Thank you for that. Yes, there is a plan to extend monitoring. In fact, it's already all across the Roatan Island. And in 2015, it expanded to other Bay Islands, such as Utila. And we're hoping that this year or next, it'll expand to Guanaja. So that covers all 3 Bay Islands in Honduras. But furthermore, because we've seen that it's been so successful, we have been sharing this a lot in very different ways with all the local actors. And what we were able to do was establish a regional water quality monitoring project in the whole MAR region in collaboration with Healthy Reefs Initiative and the Summit Foundation. So now we have 90 sites all across the MAR region. So sites which are mainly tourist sites. So we're talking Mexico, Belize, and Honduras, where there's most of the tourism comes from. And then we also have control sites where tourism is low or it's well contained with sewage treatment plants like in Roatan. And what we're doing with this project is that we really want to see how after the COVID pandemic hit and we had lockdowns, we saw that tourism went down to almost 0. And now it's been gradually increasing. So we want to track that increase and see the effect on water quality as well as reef health. And then we also want to make sure that we have this very detailed in both a spatial and temporal scale data set of water quality monitoring. So we can target our areas of where we want to apply new solutions to water quality and maybe implement these sanitation best management practices there. Does that answer the question?
Thank you, Antonella. The question for Bob. In a presentation yesterday, the reef data from areas of Florida continue to show a decline in Goliath grouper, where the Marine Recreational Information Program data seem to show a recent increase. Have your data on juveniles shown a change in abundance? Are there too many?
So the abundance of juveniles inshore is extremely variable. We actually attempted to have a third site for this, the Caloosahatchee River, which is sort of the West Coast equivalent of the St. Lucie, where they also, that's the, also receives water from Lake Okeechobee that's channelized down the river. And we really struggled to tag more than a handful. Whereas if you go down to the 10,000 Islands area, which is the center of abundance for the species, is really Southwest Florida. If you go into that habitat, everything you throw at a mangrove comes back. Every other cast comes back with a juvenile Goliath grouper. So, in terms of whether or not we're seeing a change in abundance, I believe that so the best source of data we have for that is the Everglades National Park Creel Survey. I believe that has the most recent data is showing an increase in juveniles. The concerning part, of course, is that the adults don't seem to be, that increase doesn't seem to be carrying over to the adults. There's many reasons for that. There is a extensive catch and release fishery here in Florida. And we know that bycatch mortality is non-zero. We haven't been able to adequately capture that. But certainly adults are not increasing as fast as the juveniles are. There's also the susceptibility to red tide that I mentioned as well. So it's difficult to quickly answer your question, I think. But certainly we've found that in the right habitats, the juveniles are quite abundant of the species.
Okay, thank you, Bob. And we do have another question for Lizzie as well, from Antonella. Did you see any changes in time in natural reefs after the establishment of the artificial reefs? Things such as reduction of herbivores or increase in macroalgae?
That's a really important question. There's actually been very little to no research on recruitment or problems with artificial reefs and their natural reefs. There's some study on the artificial reefs and how their recruitment is, but very little on the effects of the community that they're deployed in. Regarding the reduction of herbivores or any of those other changes in the natural reef, part of this study that was funded by FWC is actually for non-consumptive effects of artificial reefs. So looking at the grazing assays of parrotfish, and we saw a very drastic but still localized impact on how the parrotfish behave and how with the increase of predatory fish like barracudas, but it decreases very significantly at that like 3 kilometers away from the artificial reef. But as I said, there's no consistent studies on what happens when you put an artificial reef in, and that's something that we really need to look at for future artificial reefs. There's a huge lack of monitoring for any artificial reef. They just kind of throw it in the water and they're like, divers enjoy. And what we're saying is we need to have management goals and a plan to look at how it's affecting the area.
All right, thank you, Lizzie. Well said. So we are coming up on time with our session ending at 11:30. So let me go ahead and say thank you so much to all our panelists for sharing their exciting work and very thoughtful answers to many great questions for our audience. So thank you for that as well. I do want to thank everyone for joining this session for a very productive conversation. I'm super encouraged by all the great research that's being conducted and everything that we've been hearing today. For more information, please take a look at everyone's great pre-recorded presentations that are available on the conference platform. And I've also seen that our presenters have been quite responsive to questions about their recorded presentations through the virtual Q&A option on the Hove app. So please continue to reach out. And thank you so much for a very enjoyable conversation.
Thank you, Tony.
Thank you too.
Thanks,
you
Tony.
all.
Thanks, everyone.
Thank you, Tony,
Thanks,
and thank you to
everyone.
all our wonderful panelists today. It was really a great session, and Tony did really well to keep us exactly on time. So thank you again, Tony, for your wonderful moderation today. We're just going to take a quick break, just a 15-minute break, give our interpreters who are doing an amazing job today a little rest. During this break, I'm going to be showing some short films, so please do enjoy, and see you in a little bit.
Thank you.
The Florida Keys has long been recognized as the birthplace of flats fishing. Beginning soon after World War II, anglers began fishing the Florida Keys for bonefish, tarpon, and permit. In the world of flats fishing, the Florida Keys is hallowed ground. It's the home of most flats fishing world records and is on the bucket list of anglers from around the world. From those beginnings, the flats fishery now occurs throughout the region. Recent economic studies in the Florida Keys, Bahamas, and Belize reveal that the fishery is worth hundreds of millions of dollars annually. Unfortunately, there are many threats to the fishery in the Florida Keys and throughout the region. These threats include habitat loss and degradation from coastal development, dredging, shrimp farms, and other human activities. Declining water quality introduces too many nutrients, as well as pharmaceuticals and other contaminants into coastal waters. Nutrients cause algae blooms that kill seagrasses and damage other habitats. Florida Keys was declining, so they banded together to create Bonefish and Tarpon Trust to understand and restore the fishery. Recent interviews with fishing guides and anglers underscored those concerns by showing a decline in the number of chances at bonefish during a day of fishing since the 1970s. Research since BTT's creation has greatly improved our understanding of bonefish biology. We have many of the dots figured out, but we can't yet draw the lines that show us the full picture of how to conserve and restore the bonefish fishery. The Bonefish Reproduction Research Project is aiming to fill important knowledge gaps. Exactly where do bonefish spawn? We have documented pre-spawning schools inshore, and we know they spawn offshore, but where and how deep? It's a big ocean. We also need to learn about their physiology so we can understand what makes them tick. How do so many bonefish scattered over such a large area know it's time to migrate to spawn at the same time? After hatching from the eggs, what is the behavior of bonefish larvae? And how does their behavior influence the way they are transported in ocean currents? We are also raising bonefish in captivity to help us understand their biology. We want to complete the drawing so we can devise better conservation measures to protect the fishery for future generations. Although scientists have been making steady progress increasing our knowledge of bonefish, new sources of support have recently increased the rate of research. In 2018, we had the good fortune to be approached by the Fisheries Research Foundation with an offer to use the motor yacht Albola to assist with this important research. Because of the crew and fleet of boats, the Albola enables us to spend a week or more conducting research 24 hours a day in remote locations. We use shallow water boats to survey the flats in search of bonefish schools. When schools are spotted, we use our seine net to encircle the school. Once the fish are in the net, we measure and tag the fish and record the date and location where they were tagged. When a fishing guide or fisherman catches a tagged bonefish, they report the information to our website. So far, nearly all of the bonefish recaptured were within 1 mile of where they were tagged. We also determine the sex of the fish and whether they are getting ready to spawn. Soft pressure on the abdomen can induce a male to emit sperm. For females, we use a process called cannulation. If we get eggs, they are saved in a vial and put on ice for later analysis. We also take blood samples from females. More on this later. Although bonefish have small home ranges for much of the year, our tagging information has revealed that they migrate long distances to spawning locations. These migrations can be more than 70 miles each way. The Albola has been especially helpful as we study the bonefish spawning process. We anchor the yacht near a bonefish pre-spawning site where we study schools that can number more than 10,000 bonefish. We do this by quickly catching fish with hook and line. We then surgically implant a tracking tag in the bonefish while still on the deck of the flats boat. This allows us to quickly release the fish back into the school so it won't be eaten by sharks. Just before dusk, we switch to a larger boat. From this boat, we use a hydrophone to listen for sounds that bonefish might make as they get ready to spawn. We also pilot an underwater drone to observe bonefish behavior. This is an aerial view of the underwater drone next to the bonefish school. And here's the view from the underwater drone. At dusk, the bonefish begin to move offshore to spawn. We use special equipment called a VR100 that allows us to hear the pings from the tracking tags. In the foreground, you can see the blue handle on the pole that holds the hydrophone that listens for the tag underwater. The tracking continues through the night. The tags also record the depth and water temperature where the fish are swimming. Even as we track the fish using the VR100, we are also able to see the school on the sonar. This helps to verify the tag readings. You can hear the scientists call out the readings from the VR100 as we watch the school on the sonar.
Okay, we're at 127.6 meters, 26 degrees Celsius. Now at 128.2, 129, 26 degrees Celsius.
On a recent research cruise, we tracked the bonefish swimming to 137 meters or 450 feet deep. This computer animation shows the dive. The brown at the top of the video depicts the island's shoreline. The blue background is the steep wall dropping to thousands of feet deep. This is the first time that a fish that lives in shallow water has ever been recorded diving to such depths to spawn. This is truly an amazing discovery. Back in the lab, we have a lot of work to analyze the samples we collected from the field. Remember that we collected blood from female bonefish. We use the blood to examine how the concentrations of bonefish hormones change over time. After collecting the blood, we place the tubes in a centrifuge. A centrifuge is a machine that spins the vials at a very fast speed and separates the plasma from the rest of the blood. The plasma is clear and contains hormones. We use a pipette to extract the plasma from the rest of the blood sample. We then pipette the plasma into wells in a microplate tray. We place the tray into a microplate reader to determine the concentrations of hormones. We are examining two hormones, estradiol and testosterone. This graph shows the difference between lower hormone levels on the flats compared to higher levels in bonefish in the pre-spawn school. This will help us determine the readiness of bonefish to spawn in the wild and in captivity. Remember that we also collected eggs from females on the flats. After we collected the eggs, we put them in a vial and a cooler on ice and transferred them to a freezer. The next step is to analyze the contents of the eggs. To do this, we first grind the eggs using a special machine called the bead ruptor. We put the eggs in a vial with small beads and a special solution. Then the machine shakes the vials. This breaks up the eggs so we can examine their contents. Our goal is to see how many lipids, or fats, are in the eggs. The more lipids, the healthier the eggs, and the greater the chance the baby bonefish that hatch will survive. After breaking up the eggs, we put them in new vials and then put them into the centrifuge. This separates the lipids from everything else. The next step is to use a special process to concentrate the lipids even more, using nitrogen to help evaporate the liquid. The remaining concentrated material is carefully put into a tray that contains a series of tubes. These are then analyzed by an iatroscan machine, which gives us a readout of the results. The results show the percentage of lipids in the eggs for each female bonefish that we sampled. In general, healthier females will have more lipids in their eggs, which leads to a healthy next generation. In human terms, think of it as healthy females don't need supplements. Now that we know where bonefish gather to spawn, and we know their biology, we're using the Albula as a platform to take the next steps. We want to spawn bonefish in captivity so we can study the growth of young bonefish. Remember when we caught the bonefish from the pre-spawning school, tagged them, and put them back into the school? We also kept some of those fish and took them back to the Albula. Once the fish were in the tanks, we checked to see if they were ready to spawn. We cannulated the females just like we did on the flats. If the eggs looked close to spawning, we would then give them a hormone injection to help the process along. A few hours later, the eggs were closer to being ready. We then strip spawned the female by applying pressure to her abdomen. We fertilized eggs using a spawning ready male. We added water and stirred the eggs and sperm to make sure that the eggs were fertilized. And we placed the fertilized eggs in special tanks that slowly moved the water in a circular pattern. These are the first ever images taken of bonefish embryo development. You can see the embryo develop every few hours with each image. After 24 hours, just before hatching, the bonefish embryos release enzymes that start to weaken the egg wall. Then the embryos begin to shake to break out of the egg. This is the first time anyone has documented bonefish embryos hatching. Once they hatch, they don't look anything like a bonefish. Instead, they look like a clear eel. This is called a leptocephalus. This video shows hundreds of newly hatched bonefish at the water surface in one of the hatching tanks. Their development is amazing to watch, from small larvae with no eyes and barely able to swim, to actively swimming and searching for food. We have been able to raise larvae for 8 days, which is the first time anyone has been able to do this. Overall, it takes bonefish about 2 months for the larvae to completely develop, like this one, which was filmed in the wild. Soon after, the larvae change shape into a miniature version of their parents. As we learn more about bonefish larvae, we will be better equipped to understand how different populations are connected.
Hello, everyone. I just want to invite, I'll post all these films that I'm showing through the breaks on the app. Just going to get everyone set up for our next session on invertebrates. I see Rich is here. Let me just double check that everyone is here for their session. Yes, Megan. See, there might be one or two people that aren't here, but they'll be presenting later. So Rich, if you're okay, you can go ahead and get started and I'll follow up with those that are missing. Thanks, Rich.
All right, and thank you. Welcome everybody to the session on invertebrates and aquaculture, opportunities for restoration and management in a changing climate, or not, I guess, we'll find out. Our first presenter is Megan Davis, who is professor at the Florida Atlantic's Harbor Branch Oceanographic Institute, and she will be presenting today on a Fishers-operated pilot-scale queen conch hatchery and nursery facility for restoration and sustainable seafood supply, climate change and world peace in Puerto Rico. I'm sorry, world peace and climate change, we're not in there. So Megan, if you're ready, go ahead.
Okay, thank you. Nice to see you, Rich. Thank you all for listening to the presentations today, and thank you to the organizers of GCFI. It's an honor to be able to present today. So I'm about to talk about our hatchery that's in Puerto Rico. It is funded by the Salton Saul Kennedy NOAA Fisheries, and we have several partners. Myself, which I'm with Florida Atlantic University, Harbor Branch Oceanographic Institute, and my colleagues from Conservación Conciencia, which would be Raimundo Espinoza, who's the executive director, and also the Naguabo Fishing Association with Carlos Velasquez and Julio Ortiz. Carlos is the president and Julio is the fisher and secretary. We work very closely with the fishers that are part of this fishing association. There's 22 members that are part of the fishing association. And our hatchery is located inside the fishing association in Naguabo, which is about 1 hour to the southeast of San Juan. The project began in 2019, and we did have a pause in 2020 due to the pandemic. In 2021, we were able to travel to Puerto Rico and we started installing our hatchery, and the operations have now started. They actually started with the first hatch in June of 2021 this year, and we are continuing to raise conch in the hatchery. The fishers and the community are helping us with the operations of the facility. And the fishers do collect very small portions of egg masses from the conch from their fishing grounds. Here you can see in the slide our hatchery. We have our hatchery manager is standing by the larval tanks. That's Victoria Casar. And then next behind her is the egg incubation system where the eggs are brought in and they incubate for 4 days after they hatch in the larval tanks. In fact, we just had a hatch last night. They swim in the larval tanks for about 21 to 28 days, and then they metamorphose, and that's the next section of the hatchery. And then we have the microalgae section of the hatchery. At the microscope there, you can see Raimundo, and I'm in the background working with the microalgae. So we have a hatchery that's sized to produce 2,000 conch for up to 8 centimeters. So we have a nursery system on the outside of the hatchery, and those conch will be put in to grow out coastal benthic pens for acclimation and then for restoration or for sustainable seafood. We have an English training queen conch manual that's available, and we just finalized our Food and Agriculture Organization Spanish version, which should be released any moment now. So excited about that. The project significance is that we do provide diversified incomes. The fishers are paid to work at the hatchery. We have training career opportunities for students and community members. We have also a transfer of knowledge and technology to others in Puerto Rico and elsewhere in the Caribbean. for more information, please watch our full presentation that's on YouTube. And also you can follow us on Instagram at Queen Conch Lab. Thank you.
Thank you, Megan. And I'm gonna, as has been for the other sessions, I'm gonna ask everybody to hold their questions until after all the presenters. I would prefer if you put your questions in writing in the Q&A button down at the bottom of your screens, because I'll go to that first, and then I'll go to anybody's written questions in the chat. I'll read those off later when we get to the question and answer time. So thank you, Megan. So our next speaker is Elizabeth Dormit-Moreau, who is a PhD candidate at the University of Florida. And she'll be reporting on how do stone crab Menipia mercenaria fishing practices impact disease through trophic ecology. Elizabeth, are you there?
Yes, I'm here. Can you hear me?
Yes, very well. Thank you. Go ahead.
Awesome. Thanks for the intro. Yes, I'm a PhD candidate, and this is some preliminary results from one of the chapters of my dissertation. So for anyone who isn't familiar, the stone crab fishery is a trap-based fishery with the bulk of catch being landed in Florida, but there are some small fisheries in other states and other areas of the Caribbean. And this fishery is generally a claw-only fishery, so claws are removed and crabs are returned to the water alive. The idea here is that most of the meat is in the claws and crabs are able to survive and regenerate lost claws. Regenerated claws are initially small, but through subsequent molts can grow to legal size and ideally re-enter the fishery. However, there are some known issues with this process. And the fishery itself. It's overfished in Florida. Crabs also lose access to common prey items. Mortality after claw removal is high, and re-entry to the fishery appears to be pretty low. But there are also many unknowns, and two of these fueled my objectives for this study. So first, I wanted to know what parasites are present in the fishery, and then how does prey access change nutritional condition and trophic ecology? So I sampled 2 locations in Florida before the fishing season began, mid-season and post-season in 2019 to 2020. And then using necropsy and histopathology and molecular diagnostics, we actually found relatively few parasites in stone crabs compared to a lot of other fished crustaceans, especially blue crabs, which share habitat with stone crabs. But we did find a stalked barnacle on the gills of stone crabs, which you can see in that top left photo. It was present at about 35% in Cedar Key, our northern site, and just 2.4% in Harbor Key, our southern site. We also found a low prevalence of hematodinium, which is the cause of bitter crab disease, and very important to fisheries managers, and can be transmitted between crustacean species. So it's important we keep an eye on that, as it's just the second report of hematodinium in stone crab. Then as for a nutritional condition, which you can kind of see my graph on the bottom left, we found that intact crabs have better nutritional condition than one-clawed and clawless crabs. This makes sense, as one-clawed and clawless crabs we know have reduced access to some prey items, so that is affecting their nutritional condition, it seems. And then in terms of trophic ecology, there were no clear relationships between the number of claws or regenerating claws and trophic status, but we did see that negative relationship between carbon and nitrogen stable isotope signatures. So the base of the food chain is apparently different for crabs feeding at slightly lower trophic levels. So I have a bit more of this stable isotope data coming in and some more analyses to do to get a better picture of what's going on here. But I'm excited to chat and try to answer some of your questions. And if we don't get to talk about anything, you can feel free to email me at the email address here on the slide. Thanks.
Thank you very much, Elizabeth. This gives people a little chance to check out your email address. While I introduce the next speaker, the next speaker is going to be Catherine Rose, also a student at the University of Florida and also working on stone crabs. And the title of her presentation is The Impact of Trap Confinement on the Condition of the Florida Stone Crab Winnipeg Metro area. So Catherine, if you're there.
Hi,
or,
yeah,
excuse
can
me,
everyone hear
Kate.
me?
Yes, you're coming in fine.
Yes, perfect. Hi, everyone. Yeah, so my name is Kate Rose. I am a master's student at the University of Florida, coincidentally studying under the same advisor as Liz, studying a very similar topic. And I was glad to see that, you know, she addressed a lot of the main concerns that managers have about the stone crab fishery in its current state. You know, even though in that stone crabs are meant to regenerate their claws, they're not necessarily re-entering to the fishery the way we would maybe like to see them. So personally, when I like to explain how fishing in the stone crab fishery works, I like to sort of break it down into three parts. Stone crabs enter traps, their claws are removed and they're sort of handled. If they're not of legal size, they're released, and then all of the crabs that have their claws removed are then released. And like Liz talked about, we know that claw removal can have a really high mortality. There are impacts of sort of large wounds associated with improper claw removal. And then when they're released, their feeding and fighting behaviors aren't quite the same because they're energy limited based on the prey that they have access to without those claws. What we don't have a lot of information on at this point in time is the first part of that process is sort of getting into the traps and that crab's experience in a trap. And my research was sort of aimed at determining whether or not that time spent in traps was going to exacerbate the release mortality of fishery further in the process. So I collaborated with the Florida Wildlife Research Institute, and we conducted 4 trap studies, 2 in Cedar Key and 2 in Marathon, which were sort of meant to represent the northern and southern limits of the fishery respectively. And we did those at the beginning of the season and in the middle of the season. And simultaneously, I conducted some starvation experiments in the lab so that I could compare the condition of crabs in each setting. And what I found was that crabs do have a very slight decrease in nutritional condition with longer soak times in the field. So we soaked those crabs for 7 days, 14 days, and 30 days. And the longer soak times, we did see a decreased nutritional condition. However, that relationship wasn't statistically significant. And then when you consider that some of the neurological indicators that I looked at, I was seeing no impact. That was ultimately good news for the crabs because it meant that even though potentially the crabs were declining nutritionally, that they could potentially recover. And then I also observed in comparing those starvation and those lab experiments that almost all of the crabs that we observed in the field were very different. Were in very different conditions than crabs that I had kept and starved completely, so fed them no food at all in the lab for 30 days. So again, that's good news. And it also suggests that potentially there's something in those traps that is sustaining those crabs for the short term, considering some of those crabs were soaked for 30 days, and it appears as though they're eating at least something. However, claw removal and regenerating another claw has been demonstrated to be a really taxing process, something that requires a lot of energy. So even though we're not seeing super large gaps in nutritional condition sort of in the soak times immediately, claw removal might exacerbate that. And it's something that could be really important to the fishery going forward.
Okay, thank you very much, Kate. Again, we'll be addressing all the questions after all the speakers. So type in your questions into the Q&A, preferably, or the chat, and we'll get to them. Our next presenter is Wilmeli Cruz Morero, who's now a fisheries biologist with NOAA, and she'll be reporting on in situ habitat assessment of mesophotic queen conch in the west coast of Puerto Rico.
Hello. I'm very excited to be presenting this research. As Appledorn said, I'm a new fisheries biologist at NOAA Fisheries in Chesapeake Bay. And this research was my last project of my dissertation, and I graduated during 2020, so I didn't have a chance to present it to the public. To give you a little background of this research, in 2017, Hurricane Maria hit Puerto Rico, and that caused a lot of changes in the distribution of queen conch. Based on conversations with local fishermen, they couldn't find conchs in the shallower areas where they found them before, so they went into the deeper areas to get their catch. So these deeper areas range between like 25 meters to 45. So we took that information to see what was in that area using the camera sled. We did 5 hectares with the camera sled, and we accounted everything and environmental conditions. So our overall results, we found 426 conchs, and half of them were alive. We saw signs of fishing across all the areas. But I think one of the most important things that we saw is that we found about 5,000 juvenile conchs, which is the picture right in the bottom. These little juvenile conchs are really hard to identify. So right now we're working on better photometry techniques to try to identify these little conchs using their spines. We also found egg masses and conchs copulating in the area. These conchs are very different from other conchs, so it was really interesting to see the different habitat where they are. The overall results of this project, it shows that the distribution, like conchs were juveniles and smaller conchs rather being in the 25 meters area. And there are some combinations. We also identified conchs by juveniles, newly mature, and all adult. Just visually, we couldn't take any width. So these are just visual estimations. And it seems like older conchs were mostly found in the deeper areas. Just to conclude, these juveniles have not been documented before, and we don't know the role of these mesophotic conchs. In my personal opinion, I think these are stuck on their own, but there has to be more research done to see if they're actually having a connectivity with the shallow water. It is important to know that what is like account the amount of conchs in this area, and maybe change the management regulations to see their to see if they're stuck in their own. And with that, I'll take questions later. Thank you.
Thank you, Wilmeli. I'm sure you're going to get some questions on this because I'm going to ask some. And it was a very interesting talk. The last talk for our short presentations now is by Miguel Mateo Sabedo Itza, who is a biologist working in the Mexican Caribbean. Has worked on a wide variety of projects. And today he's going to be talking about a characterization of the queen conch artisanal fishing prior and after the 5-year ban at Chinchorro Bank Biosphere Reserve in Mexico. So Miguel, are you there?
Sí, aquí estoy. Buenos días.
Okay, go ahead.
Ok, bueno, buenos días. Les agradezco a todos. Como bien dice Richard, el tema es caracterización del carácter la pesca artesanal de caracol rosado antes y después de una veda de 5 años en Banco Chinchorro, en el Caribe mexicano. Este trabajo se realiza en México. El único sitio donde la pesca legal de caracol rosado es permitido es en Banco Chinchorro, y este se establece mediante cuotas de captura cada año. En Banco Chinchorro únicamente 3 cooperativas son las que pueden extraer el caracol rosado, y lamentablemente desde 30 años atrás estas cuotas han bajado drásticamente, a tal grado de que a partir del 2010 las capturas o las cuotas de captura han bajado a únicamente 9 toneladas por año, repartidas equitativamente a cada cooperativa pesquera. A raíz de eso y ante el declive de la población y por impulso de los pescadores, se impuso o se propuso una veda de caracol rosado durante 5 años, que esta fue efectiva entre el 2012 a 2017. Este trabajo hace una descripción de la pesquería de caracol rosado, con datos con monitoreos que hicimos en el 2011, que fue la última cuota de captura de caracol rosado antes de la veda, y nuevamente hicimos el monitoreo en 2018, que fue durante la segunda captura de caracol rosado después de la veda de caracol rosado. Dentro de los principales resultados podemos observar que, para empezar, antes de la veda se detectaron más de 14 sitios de pesca que los pescadores utilizaban para poder capturar el caracol rosado. Después de la veda, en 2018, estas zonas disminuyeron a únicamente 7, y estas se centraron en la zona centro de Banco Chinchorro. Y esto es bien importante porque los pescadores no tuvieron que alejarse mucho de su zona de donde ellos pernotan, que es en la isla de Cayo Centro. Ellos ya no fueron a lugares lejanos. ¿Por qué? Porque encontraron recurso, el recurso de caracol rosado en zonas más cercanas, en donde ellos pudieran disminuir su consumo de combustible y horas de pesca. Se observó un aumento en la captura por unidad de esfuerzo de 5 kilogramos de caracol rosado a 8 kilogramos de caracol rosado por pescador por hora. Ahora, en la parte biológica o de la población de caracol, se pudo observar que hubo una disminución en la longitud sifonal del caracol, pasando de 21.3 centímetros de la longitud de la concha a 20.02. Eso quiere decir que hubo una disminución ¿cómo se llama? Significativa. Pero ¿qué pasa con la longitud del labio? También se midió la longitud del labio, y por lo contrario, esta longitud aumentó, pasando de 3.7 milímetros de grosor del labio a 10.3. Esto es bien importante porque se puede observar que ha habido un incremento en la proporción de organismos que ya se consideran maduros sexualmente. Sin embargo, hubo una disminución del tamaño del caracol debido posiblemente a la sobrepesca. Ahora, por último, dentro de la Carta Nacional Pesquera en México, establece una talla mínima de captura de 20 centímetros de la longitud de la concha. En 2011, que fue antes de la veda, únicamente, bueno, el 22% no cumplió con ese criterio, ¿no? Fueron caracoles por debajo de esa talla. Sin embargo, después de la veda, ese criterio aumentó a 40.4%, es decir, se capturaron más organismos por debajo de esa talla mínima de captura. Dentro de las principales conclusiones tenemos que la veda de 5 años sí fue una estrategia efectiva, parcialmente, debido a que se pudo observar una recuperación del caracol rosado. Sin embargo, aún permanece afectaciones, que es la sobrepesca, y además de que los pescadores vieron no tan efectiva la veda porque ellos esperaban que la cuota incrementara, ¿no? Pasado los 5 años. Sin embargo, esa cuota ha permanecido únicamente a 9 toneladas anuales. Gracias.
Okay, thank you, Miguel. I see we do have some questions, and I'm actually going to use my position of authority here to actually start off with a question, and this is to Megan Davis. Megan, I'm interested in how the electricity costs for the hatchery are being covered. I know that in Puerto Rico that municipalities, at least at the moment, get free electricity. And so I was wondering whether this facility is considered a municipal facility and you're getting free electricity for all your pumps, or whether this is a cost you've had to build into your project.
Thanks for that question, Rich. So the electricity is my understanding that it's not covered as free, but I do want to let you know that the Fishing Association is using solar power. And so it is almost 100% solar power. And so part of our hatchery is on solar, and we've just purchased more solar panels so that we can be 100% on solar. So we're looking forward to that. Thanks for the question. Thank you.
Two questions for Elizabeth. And these are from Michelle Shar. How long does it take to regenerate one claw? And secondly, what are the main differences between prey availability to two-clawed versus one-clawed versus zero-clawed crabs? And how does that translate to their nutrition and potential regeneration rates?
Thanks. Yeah, great questions. That I actually did a lot of work related to these things for my master's. So how long does it take to regenerate a claw? Kind of depends on where in the molt process the crab is when you take the claw, but in general, these crabs are adults in the fishery, and adult stone crabs molt once per year. So within a year, you're going to get a tiny little regenerated claw, and a year later you'll get a slightly larger one. Basically some modeling and some experiments I did show that it's going to take like 2 or 3 or more molts for a crab to get a claw big enough to re-enter the fishery. And that's a really tight window because crabs enter the fishery at about 3 years of age, and they senesce and die at 8, we think like 8 years maximum. So there's really not much of a window for them to regenerate and re-enter the fishery. And then, so what are the main differences between prey availability? That's going to be bivalves. So bivalves are, we think, a huge portion of stone crab diet. They are scavengers, so they'll eat anything they come across. But they've got those huge claws to crush the bivalve shells. But when they lose one claw or one claw is removed, they still have access to bivalves. They can still use their pincher claw. So they've got a crusher claw and a pincher claw. Pincher claw is smaller, but can still be used to either like pry open or sometimes crush the bivalves. We think it takes a lot longer for them to do that. And then when you take both claws, they basically don't have any access to the bivalves. So they're reliant on any like softer or easier access prey items that they find. And we haven't done any direct experiments to answer the second part of your question to see exactly how those differences translate to their nutrition and potential regeneration rates. It's kind of what I'm trying to get at with my field experiments and collecting crabs that have been missing claws. It's kind of hard to do those direct experiments because it takes so long. Well, first, so many crabs die as a result of claw removal, and then it takes so long to like get them to regenerate claws. You've got to keep crabs in the lab for a year plus. But we think basically that, you know, any reduced prey availability is negatively affecting their nutritional condition and then probably slowing down their regeneration.
All right, thank you, Kate. Or Elizabeth, sorry. Because the next question is for Kate. It's from Peter Randall, who says, interesting presentation. Have there been any comparisons of survival rates between crabs released with claws and those that have had their claws removed?
Yeah, so for my study, I chose just to look at sort of that short-term nutritional condition, because as Liz mentioned, sort of, and in some of the work that she's done in her past, in order to sort of observe survival, you have to keep them for, you know, a year or so, or until they die. And I was conducting these experiments in the height of the COVID-19 pandemic. So it was sort of challenging to keep them in an environment and keep them healthy and keep myself healthy and everybody around me in the process. But I might actually defer to Liz on that question, since she's done a lot of work in this arena.
Yeah, so I personally have done some experiments with claw removal and survival in the lab. And what I found in the lab was that the biggest factor in survival is actually the where the break occurs. So there's a natural fracture plane on the stone crab, and if you get it, if you break the claw right on that natural fracture plane, they basically don't lose extra muscle and extra blood, and they have a much higher chance of survival. That being said, there was definitely higher mortality for one claw removal than, you know, crabs that you didn't take any claws from, and then higher still mortality if you take both. FWC did a great study where they actually took claws and put them in traps, but in nature. So they could see that there was some mortality from their intact crabs, but like I said, higher mortality. I think it was about 13% mortality for intact crabs, 40% for when they removed one claw, and then 60+% when they removed both claws. And then the other interesting thing they found was that this was really temperature dependent. So in warmer locations and warmer seasons, mortality was a lot higher.
All right, thank you very much. Next question is again from Michelle, and it's now going to be directed for Miguel. And Michelle writes, ¿Esta veda fue acatada en toda la zona o en qué por ciento se cumplió durante los 5 años? Sí se sabe.
Hola, sí, yo creo que las dos preguntas, tanto la de Strader como de Alfonso Aguilar, las puedo contestar, creo que a veces las puedo contestar ambas. Sí, desde que se implementó la veda por 5 años, Chinchorro es bien conocido que es una zona de alta pesca furtiva. A los pescadores furtivos aquí se les denomina localmente como pachocheros. Son los pescadores que llegan y sacan el caracol rosado de manera ilegal. Durante la veda de caracol rosado, durante los 2 primeros años, sí hubo una vigilancia y hubo toda una estrategia para poder controlar la pesca ilegal. Y esto se vio con otros trabajos que hemos realizado en más cuestiones de densidades y tallas. Dentro de la población se pudo ver que sí hubo una recuperación significativa durante los 2 primeros años de la veda de caracol. Sin embargo, desgraciadamente, esta vigilancia pues bajó para el 3º, 4º y 5º año, lo que provocó que la pesca furtiva incrementara dentro de Banco Chinchorro. Y lamentablemente todo ese esfuerzo que se hizo durante los 2 primeros años, pues se vio mermado, se vio disminuido. Hay estimaciones de que normalmente, bueno, anualmente hay una captura ilegal de caracol rosado que va desde las 32 hasta las 320 toneladas de caracol rosado, ¿no? Anuales solamente en Banco Chinchorro. Platicando con pescadores recientemente que fuimos a Chinchorro, ellos nos comentan que aproximadamente son 10 embarcaciones furtivas que hay en Banco Chinchorro de pescadores, y que ellos no toman en cuenta la longitud sifonal, ni grosor de labio, ni zonas de recuperación pesquera. Ellos van y extraen todo lo que puedan, ¿no? Esas 10 embarcaciones casi dan como para otra cooperativa de pescadores. Sí. Entonces, lamentablemente, aunque sí la veda tuvo un impacto positivo dentro de la población de caracol, si se hubiera seguido con la estrategia de vigilancia y todo, posiblemente hubiera tenido mayores resultados.
Thank you. I might add, at least looking at the data in your presentations, that perhaps a closure more than 5 years would also been necessary, because it looked like you were starting to get recovery, but you know, it takes 4 years to get your adults in the first place, so that doesn't leave a lot of time before you open the fishery to really accumulate biomass. So it's interesting. We have a question now for Team Stone Crab from Joanna Pitt, who asks, so if it seems likely that a given crab might only produce one extra claw over its lifetime, would it make sense to just increase the minimum size so that individuals are taken at age 6 or 7 and sacrificed at that time so that both claws can be harvested? Same harvest potential, but healthier reproducing crab up until that time.
Do you want to
So,
start,
Liz?
Kate, or you want me to?
I think I would probably would have, sure, I'll take that. And I probably would have started with, I think this is a challenge in the stone crab fishery, is the modeling in general that comes with it, because when you don't, you know, when you take a whole animal, you know what happens to that animal. Whereas when you're taking just the claws off an animal, you don't know how long it's going to live. And despite the fact that, you know, we sort of addressed that there's a lot of release mortality and they're not potentially regenerating, re-entering the fishery, there is some potential that those crabs contribute to the population still. They still reproduce. So it gets a little muddled when it's kind of hard to recommend regulations. I would say, I know that there are some researchers at the University of Florida that are trying to sort of tease out the intricacies of that. I don't know, do you have anything else to add, Liz?
Yeah, that's spot on. And I would just add that there has in Florida been a recent increase in size, the size limit. So it's not to the point where we're taking like already senescing, you know, 6 or 7 year old crabs. It's a much smaller increase to not get too much anger from the fishers, I think. But there are that and other ongoing changes to the fishery in Florida.
And I know I was thinking also a regulation. I think one of the things that I've read a lot in the literature is that when it comes to reproduction, there seems to be a limitation of large males in the fishery, because the size limit that is in the fishery, you know, it was 2 3/4 inches, and now it's moving to 2 7/8, allows female crabs to reach maturity and even contribute to the population a couple times before they enter the fishery, whereas larger males or males don't mature before they reach the fishery. So taking those larger males out of the population, it could help, you know, prevent or give some sort of you know, a shield to those larger crabs as well, and help them reproduce more.
Okay, thank you. This is another question on the crabs going to Liz. This is from Ron Hill. It says, your info is really interesting. The stone crab fishery has been characterized as very sustainable since they were only taking one claw and it grows, quote, right back, unquote. The fact that it takes so long to regrow totally refutes this impression. Should your information change the management regulations of the fishery?
So I will say that managers have known this information that Kate and I have been chatting about to you guys. They've known this for a while, that crabs aren't really re-entering the fishery. So it's not so much like brand new information. It's part of it is getting like the details of how many crabs are dying, how many are surviving, and also talking to you guys about it. Like I have really found in speaking to fishers and residents, especially in Florida, that there's just this whole mythology around the fishery and how wonderful it is and how sustainable it is. And that's one of the parts I really enjoy about my research is getting, unfortunately, it's like not the greatest news that I'm providing of this wonderful fishery, but being able to talk to people and get that information out there. So like I said, I don't think that this stuff has been unknown to managers, but at the same time, as Kate mentioned, there are researchers doing some stock assessments and trying to get more specifics to the managers. And there have been several regulation changes over the past few years. Also, I did my master's work in South Carolina, which has a very small stone crab fishery compared to Florida, but my research did inform managers there to keep they have a one claw removal fishery, whereas Florida allows removal of both claws. And my research did, convince them to keep it a one claw only fishery.
I just wanted to add to sort of the conceptions that Liz had talked about talking to members in Florida. I had a really interesting conversation with a few people the other day. I was out in Cedar Key where I conducted my lab experiments, and they said, Joe's stone crabs, which is a really large not manufacturer, but distributor of stone crabs, not only for Florida but throughout the state. One of their big marketing points is we only take one claw and that's better. So a lot of people in Florida weren't even aware of sort of the regulations that you could take two. So it's sort of having to meet the managers and the fisheries and the public and get them sort of all in the same arena.
Yeah, I've heard a lot of interesting stories along those lines too, Kate, that I think there is a bit of like a purpose for that. We only take one claw because it's better, and it keeps a lot of recreational fishers only taking one claw versus both. But I've also heard this myth that taking two claws actually lets the crab regenerate faster and re-enter the fishery faster,
or that they won't get into as many fights. They won't fight with other crabs because they know that they have no arms. I've heard that one too.
Yeah, which is, not much evidence behind that one.
Okay, thank you for that stimulating crab conversation. We're going to switch over to Conch for a few questions here. Well, first, that's more or less a comment from Alfonso Aguilar, who's referring to the ban at Chinchorro. He says, so the 5-year ban did not help in that sense. And due to the high illegal fishing, which has no solution, the same problems have occurred in Alacranes Reef, where there is a permanent fishing ban. So he's making the case that the illegal fishing is a very serious problem that's affecting the ability of the conch to replenish itself. And Michelle Scherer asks of Megan, can you share any results of your hatching survival rate?
Exacto.
Sí. I'm sorry. Miguel, do you want to make a comment?
Bueno, sí, exacto. Como bien comenta el doctor Alfonso, es una problemática, ¿no? La pesca ilegal. Es complicado poder disminuir esa actividad y tal vez, y también la pesca ilegal, pero sobre todo también el consumo, ¿no? Ilegal que hay, porque es muy común ahorita con redes sociales andar vendiendo y promocionando el kilo de caracol tan descaradamente, ¿no? Y si hay una demanda, pues los pescadores furtivos o los pachucheros pues van a continuar sacando el caracol, ¿no? Y usted comentaba de poder incrementar de años el periodo de veda. Sin embargo, socialmente o la percepción de los pescadores que sí tienen el permiso, pues ellos comentan que para ellos no es conveniente, ¿no? Implementar nuevamente otra veda porque los resultados para ellos no fueron satisfactorios, ¿no? Porque ellos esperaban que su cuota de incrementara, sin embargo, eso no fue posible. y pues ellos comentan que por parte de ellos ya no quieren implementar otra veda de caracol rosado.
All right, thank you, Miguel, for those comments. It's really interesting. Again, Michelle was asking, Megan, can you share any results of hatching survival rates in the experiments compared to the natural ones in waters where egg masses are being extracted?
Thank you for that question, Michelle. So in the wild, the female is laying anywhere from 9 to 16 egg masses on the average in a given season, and that season will change slightly depending on if it's like the Bahamas versus Puerto Rico, for instance. And each egg mass is having an average of about 500,000 eggs. So in the wild, the prediction is less than 1%. So in the hatchery, what we do is we actually bring in a very small amount of the egg mass and allow the rest of it to hatch in the wild. So when we have a hatch, they pretty close to 100% of those eggs hatch. Our limitation in a hatchery is the size of our hatchery larval tanks. So we could grow more of those. We keep as many as we can, and then we release the ones that we can't grow after they hatch. So let's just say in the larval tanks that we have in Puerto Rico, which is a pilot scale hatchery, the larval tank is 68 liters, and we hatch them at around 100 to 200 per liter. By the time they're ready for metamorphosis, which is about 3 to 4 weeks later, they're at 10 to 20 per liter because they've gotten so big. So it's not so much the survival as it is the managing of the hatchery. And so whatever conch larvae we can't keep in the hatchery, we're always releasing them. So then they go through metamorphosis, which can be another challenging transition in a conch's life cycle. And about 50% to 70% of them will go through metamorphosis, depending if they're competent and ready for metamorphosis. And then after that, you're going to have some mortality around maybe 75% until they get to the size of about 1 year old. And they're still in a nursery. They're still maintained in a hatchery nursery situation. So just thinking about the metamorphosis and then the survival for that first year, you're somewhere between 30 to 40% from that original hatch, which is pretty high, pretty significant to be able to do in a hatchery. And then really once they go out into the field is when they're going to be most vulnerable again when you do the restoration. And there's a lot of great papers by Rich Appledorn and Al Stoner that have talked about the challenge, and Bob Glazer's group as well, that have talked about the challenges of restoration. And that's an area that we're going to definitely put some more focus in on, doing some acclimation, some making sure that they're behaviorally and morphologically adaptable to be able to be restored, making sure they get put into an area where there's already a nursery of conch. And so this will be the bigger challenge, is getting them from 1 year up to 3 years. And so we'll be looking at survival improvements in that area as well.
Thank you, Megan. And I'll follow that up with a question from Ron Hill. What is the ultimate goal of your project? Is there a profit potential to the project?
Thank you, Ron, for that question. So this is a pilot scale hatchery. And so the main goal is to be able to do some conservation education with the fishers and the community, along with restoration. Our second Salt and Salt Kennedy grant that we just received will also be to do some grow out in in pens in the field. And so those conch will be put into the marketplace. It's very interesting. I'm learning a lot about the culinary aspects of conch in Puerto Rico. It is sold as a steamed salad. And so we're also going to be working with the chefs and different restaurants to see if we can also improve the value of the conch. And so being able to improve the value means that possibly you don't have to fish as many conch because you're going to get a higher value. And so that's also part of how, so we're trying to take a holistic approach. But to really answer your question, Ron, the hatchery at the size it's at would not be able to be commercially profitable, but the techniques can be applied to larger scale. So we're looking forward to all different aspects of how a hatchery like this can be used and also grown over time.
So thank you, Megan. Are you also working on how to get Social Security payments increased so those of us on fixed incomes can enjoy those higher valued conch?
Okay, so that's a good question, Rich. I think when we talk about higher value, though, it just means that it's going to have, more different recipes that are available for it. And yes, you're right. It could definitely increase the conch to such a value that it's hard for everyday edible products. So thanks for that comment too.
Yeah, well, I'd like to add my comment that that's actually a really important aspect that's totally neglected in Puerto Rico, is basically what they do with the product after they take it out of the water. And there is, I think, a lot of quality loss in how products are treated, even when the chefs know what they're doing. By the time they get them, they've sometimes been just frozen and held in a freezer for a while. Conch is actually a little bit better off than, say, fish in that regard, but nevertheless, this post-processing aspect to increase the value, I think, is something that's very, very much needed. So I'm glad to see that you're looking at that.
Yeah, thank you, and thank you, Rich. And just one other comment, if you don't mind. I have been working with the fishers in the Bahamas, and they're also looking at value added of the conch shell itself, and are making some beautiful crafts with the conch shells. And I think there's a perfect opportunity for more of that in Puerto Rico and throughout the Caribbean, to be able to increase the value of the shell itself.
All right, thank you. We're going to continue with conch, but switch over to Wimeli. Again, a question from Michelle Shar, who says, great work and presentation. Can you preliminary say if the deeper areas had a different habitat composition that would favor a higher density of larger conch?
That question has a lot to think about. I think maybe one of the reasons, and I'm just assuming, is that there's a higher density of these larger conchs because they're harder to get, and like they're not as subject to the fisheries as other conchs. But in terms of like dietary and habitat per se, the habitat is quite different than the shallow waters that we did a year before this research. So the conch in these areas are adapted to be in a more rocky, and then the type of algae that what I think they will be consuming, it was just a filament of like algae across the whole area. So their diet is different and the habitat is completely different. So I don't know if that will favor it, but I think the thing that the difference between density is mostly like the pressure of fishing in this area.
All right, I have a couple of questions for you, Wimeli. Well, first a comment. Mesophotic generally has been limited to 30 meters and below. And most of the people looking at that wish it was lower still. So you're kind of pushing the definition of mesophotic the wrong direction. So you might think about that when you're going to publish this. So one comment on the juveniles. I used the kind of scientific method of staring at that photograph for a long time until truth was revealed to me. And they certainly look like queen conch to me, but they're very small. They're like under 5 centimeters, looks like. But just from the spine structure, you mentioned you're looking at the spines, and they definitely look like queen conch spines to me and not fighting conch spines. Maybe I would turn to Bob Glazer to give a comment on that as well, because he's pretty good at that. But the habitat that you showed there, where you had lots, it's all soft bottom and you had lots of trails. So when we did some ROV work on that north drop off of the western platform off of Tourmaline, we were seeing conch in 60 meters of water, adults. And while I only saw a few individuals, there were lots of tracks running around. And since I had no control of the ROV, we were on basically a current, flow with the current dive. We couldn't really check that out much, but there's lots of conch down there. And so I'm glad to see that you're able to document that. And with that, I'm going to turn to Bob's comment, who says he agrees on the queen conch assessment. Looks like adults that are, oops. I have to get the chat up here. Looks like adults that are either food limited or in very high densities. And so I don't know if you want to make a comment on that. You certainly have experience, especially using the same methodology of looking at the conch up on the platform and now in these deeper areas. So again, you say it looks like adults that are either food limited or in very high densities. Do you get that?
I don't understand the comment, sorry.
I would say it has to do with the morphology of the conch.
Oh, okay. Yeah, the morphology were definitely different than the shallow water in the previous research we did. Conch seemed thicker in this area. And the few ones that we saw, the fishermen actually got into them. So they made a little hole in the spiral. Like you can visually, like I can't really measure it, but you can see that that shell is a lot thicker.
Okay, by thicker, you're talking about the shell thickness and not necessarily the width of the conch relative to the length. Is that correct?
Yeah. Just observational. They do seem thicker, but I don't have a way to actually measure it with a camera looking down. Just an observation.
All right, I'm going to go stay with a question from Bob, who is having trouble with the Q&A tab. And this is for Megan. Megan, is there any interest producing conch for the higher value products, such as aquarium trade or even pearls?
Thanks for that question, Bob. So in regards to aquarium trade, I think it's risky to use juvenile conch for that particular trade because of the possibility of collection of juveniles from the wild. However, as we've been talking about, there are West Indian fighting conch in Puerto Rico, and the West Indian fighting conch are actually excellent to be able to breed in captivity and to raise them for the aquarium trade. So that is something that we would be quite excited to work with that species as well, for specifically for the aquarium trade, but also as a culinary dish as well. Because I understand that the fighting conch is eaten throughout different regions of the Caribbean. In regards to pearl, actually we've had a number of requests to work with us with our pearl patent, and so I'm excited that we may have found a couple of places in the Caribbean where we could work on that, and I think that has great potential for a number of places throughout the Caribbean. So thank you for those questions and comments, Bob.
Bob has a question to all conch people. Has anyone seen a decline in conch reproduction found in shallow areas? And I can say in at least my part of Puerto Rico, the answer is no, with a big qualifying caveat, and that is a lot of our inshore shallow areas have undergone a fair amount of habitat alteration, especially siltation. And so conch aren't really found too much in those habitats anymore. So that trend overrides anything that might be due to, say, temperature changes. And if anybody else has a comment to that one, chime in.
I can talk from the southeast side of Puerto Rico, where the fishers are collecting egg masses for us, that most of the egg masses are coming from 50 to 120 feet deep populations, which mirrors what Rich is saying, and also what Wilhelmine had also mentioned about the shallow water. They are finding, however, juveniles in the coastal areas, and I've seen some of that. So I'm not sure. It seemed like after Maria, a lot of the shallow water conch that were collected in the southeast side of Puerto Rico really did disappear from those areas. So there may have been some traditional, more shallow water populations. Maybe Rich, you know a little bit more about that as well.
Well, I would just comment that Maria took out a tremendous area of grass bed and just totally destroyed. So there was a lot of habitat loss that those juveniles would have been in. Judging by the amount of stuff that was deposited on the shore,
yo hablo un poquito sobre Banco
a
Chinchorro,
lot of,
en Quintana Roo, México.
Miguel, can you wait?
Sí.
Can you wait, Miguel?
¿Sí me escuchan?
Okay, so,
¿Sí me entienden?
yes, but I was still talking.
Okay.
So Maria, judged by the amount of habitat loss and burial that happened on land, probably a tremendous amount of juvenile conch were also buried probably too deep to get out. So there was probably a fair amount of mortality associated, at least in that eastern area that was impacted. So Miguel, yes, go ahead.
Disculpen, eso es la interrupción. Sí, bueno, en Chinchorro, tradicionalmente se ha monitoreado mucho la zona somera en Banco Chinchorro. Y como bien comentan, son sitios en donde actualmente ya no se encuentra eventos reproductivos. Nosotros en una capacitación y un entrenamiento que hicimos con pescadores, hicimos ciencia ciudadana en 6 sitios en Banco Chinchorro con profundidades donde normalmente no se hace el monitoreo. Profundidades entre 6 a 15 metros, que son en las pendientes, que son los arenales. Ahí pudimos encontrar eventos reproductivos, afortunadamente, muchos eventos reproductivos, cópulas, masas de huevos. Esto fue en el año pasado, en agosto, y este año lo repetimos y afortunadamente vemos que sí hay esos eventos reproductivos, ¿no? Pero en la parte somera, a profundidades como de 2 metros, 1 metro, donde tradicionalmente se encontraba, pues ahora no hay.
Okay, thank you for those comments, Miguel. Bob is also clarifying his question, the reason for the question. We are starting to see a signal that temperature may be impacting reproductive development in shallow areas. And to that, I would add, in the 2 reproductive experiments we've done in Puerto Rico, we have seen at the warmest part of the year, a slight dip in the reproductive rate. And I have, since it's so slight, I don't know whether it's actually driven by temperature or not, but it was an interesting observation and would perhaps be consistent with what Bob is talking about. So any other comments relative to Bob's question about temperature? Michelle's writing, could temperature changes also affect the timing, but not the, can temperature changes also affect the timing, not only the areas? And I think that's kind of what I was pointing out in the experiments that we had done, is that it didn't affect the timing in terms of when they started and necessarily when they finished. Temperature does seem to affect that, but it's low temperature at that point. But there might have been a high temperature impact that slowed things down for a little bit briefly in the summer.
I want to add to the temperature. I don't know, this is just an observation from part of our survey. The survey was done in July and August, and we saw conch egg masses and conch in copulating activities in the month of July. So that may be something that should be in consideration for future management practices.
Okay, thank you. Not to leave Team Crab out of the conversation. So Michelle is kind of pivoting this question over to stone crabs. Says, Elizabeth, how will the increased temperatures of climate change affect stone crab ability to support the fishery?
Yeah, great question. So many ways potentially. But the only thing that's really been studied with temperature and stone crab so far is what I mentioned earlier, as far as the release mortality is higher with higher temperatures. But as we've discussed a lot today, that may not affect the fishery so much since survival after coral removal is so low. But obviously there's tons of other ways. Increased temperature is going to increase their need for food. And when you increase, their metabolism and need for food, but decrease their access to food, you're gonna run into problems. So many other ways. Kate, do you want to chime in?
Yeah, I can add to that. There is a researcher at Moat who recently published some results from his study where he was specifically looking at how rising temperatures and potentially elevated CO2 pressures and consequently, ocean acidification could affect the larvae of the stone crabs while they're in that pelagic stage. And he found that both elevated temperatures together and increased pressure of CO2 reduced larval survival. And then there were some more intricate results as far as sort of how those two things affect their motion, so as I'm sure you guys all know, when things recruit to an area, they're using light cues, they're using chemosensory cues, they're using gravity cues. And I can't quite remember, as there were 5 larval stages and each of them had 3 treatments, but there was some evidence that those elevated temperatures and those elevated CO2 pressures, higher acidification, were disorienting the larvae. So potentially you're looking at some reduced recruitment to the fishery as well as a result of larval survival, and then also just being able to settle and grow into adults. So sort of a two-pronger there, especially with sort of all of the intricacies we discussed as far as, stone crabs, even though they might not be surviving to regenerate claws, they could be regenerating or they could be contributing to the development. So yeah, lots of variability there. Hopefully that model that's coming out of UF soon will be able to provide some more information, which would be really cool.
I also want to mention, because a lot of my work is on disease ecology, and I've been looking into stone crab diseases and parasites quite a bit, that this was part of my hypothesis that, the fishery and that climate change and that a lot of things that we aren't looking at yet could be affecting parasite transmission. And so again, with that unique nature of taking a claw and putting the crab back in the water, I thought that if you're increasing density in a stone crab trap, and you're increasing the potential for transmission of diseases. And also with climate change, there's a lot of diseases that are proliferating and popping up with warmer waters. I just thought all of these things might be compounding to impact the fishery in ways that no one's really looking at, because if you're increasing transmission in the trap and then releasing, even sublegal crabs are in that trap and we're not taking a claw from them, you're releasing them, you could be spreading diseases around. And so far, what I've found has been really interesting, that stone crabs aren't harboring a lot of diseases and parasites. So that, might have been an unfounded worry on my part, which is a bit of a positive in all of this.
Okay, thank you. And now we're going to switch back to conch. We've been having sort of a side conversation going on in the chat. So Bob is going on about the reproduction and the temperature issues, and he's saying we saw reproduction occurring in months where it never did, as far as we know, which has implications for dispersal and metapopulation stability, since our retentive mechanisms do not occur during those months. To which I'll point out that we've seen variability in the timing of spawning here in Puerto Rico, depending on whether it's been a cold year or a warm year. And you can go back to one of Randall's works where he was talking about in warm years, they were seeing reproduction almost all year round, although there's still, the main summer peak. And Bob's comment about the retention mechanism is really pertinent because of the big gyres that set up off of Florida, which are presumed responsible for bringing larvae back to the system. And I know from modeling work that's been done in Puerto Rico, there are seasonal gyres as well. They have not been looked at in terms of their effect on seeding conch locally from the reproduction that's happening here, but the systems are dynamic in that regard. And Megan also comments, that's interesting. Are the Florida queen conch laying later in the season or starting sooner, or both? And so Bob will have to type his answer. And I'll read from Michelle. Thanks, Bob. This would be key to the fishery with a static closed season that may or may not cover the whole aggregation when they are most vulnerable. And so Bob writes, they're starting sooner, but that was an observation from about 10 years ago. Not sure if it's still occurring. The reproductive impacts seem to be the range of temperatures they're exposed to. Well, not the absolute temperature, but the range of temperatures that they're dealing with. Megan also throws back likely photoperiod 2. And hopefully photoperiod is not changing with climate change. So I will ask, are there any more questions for our panelists or other comments? We're getting close to our 2 o'clock deadline, still a few more minutes. Okay, Bob is writing back. I don't think it is photoperiod because we saw reproduction on the shortest day of the year. So I would say that's a startling observation. So maybe at this point, it would be a good opportunity for us to terminate the session. And we will, let's like to thank our 5 speakers, first of all, for their presentations. And if you haven't, please go and see their full presentations that are available. They're all quite interesting. And you can have further discussions with everybody offline. And I would like to thank everybody who's been in on the session for your patience and for your good questions. So thank you very much.
Thanks, Rich. And as we get all the panelists join in for the next session, I'm just going to share a quick little video about queen conch from Megan.
Huevos?
Yeah.
See?
Take it. Take it. Is this?
Ah,
Is this?
very nice.
And thanks again to all our panelists and moderators in the invertebrates section. It was really a good and an exciting discussion and some really informative presentations. So I thank you all again for being here. And as I've said before, please don't let the discussion stop here. Please go ahead and look at all the pre-recorded presentations and continue to interact with all the speakers. So thanks again, Rich and all the panelists. And I will invite Reed to join us now as we move on to our panel discussion in the science, data, and innovation session.
Great. Thank you. Hello, GCFI friends and colleagues. It's good to see everybody. I look forward to hopefully seeing everybody in person next year. We'll see how it goes. Again, I'm Reed Hendon. I'm with the University of Southern Mississippi's Gulf Coast Research Laboratory, and I'd like to welcome you to the Science, Data, and Innovation session of the 74th GCFI meeting. This session will also include 2 presentations originally submitted for the megafauna and pelagic species session. So we will have seven panelists for this session. As with the other sessions, each panelist will have 3 minutes to give us a brief overview of his or her presentation. After all the panelists have spoken, we'll open the session up for questions. And again, we ask that you use the Q&A button at the bottom center of the Zoom window to submit those. In the interest of staying on track with time, we'll go ahead and get started. Our first panelist is Tony Marshak, who is Program Analyst and Research Associate with CSS Inc. in support of NOAA's National Centers for Coastal Ocean Science in the U.S. He is also co-author of the recent book, Ecosystem-Based Fisheries Management, Progress, Importance, and Impacts in the United States. So, Tony, take it away.
All right, thank you, Reed. Good to see you. So hi, everyone. I'm Tony Marshak. I work in support of NOAA up in Silver Spring, Maryland. And over the past couple of years, I've been collaborating with Jason Link, the senior ecosystem scientist for NOAA Fisheries, on characterizing and comparing large marine fisheries ecosystems of the United States, including the Northern Gulf of Mexico and US Caribbean, for determining successful approaches to ecosystem-based fisheries management, or EBFM. And these approaches are multidisciplinary. They consider an entire regional fisheries system together with human dimensions. We've been examining these large systems and evaluating facets of their ecosystem level management as well as their progress through a coupled socio-ecological lens, which we outline in this figure that comes from a recent paper and book. And this includes factors like the natural and human environment, including basal ecosystem production, with direct connections to the status of their living marine resources, fisheries, marine economies, and governance approaches. And we looked at data for these 90+ indicators in each US region and multiple subregions, and their interconnectivity is related to successful EBFM. We also note the relationships in our proposed pathway for a given fisheries ecosystem at the bottom of this slide, where we've observed that primary production really sets the foundation for total biomass, which in turn sets the foundation for total potential landings, and that these factors also greatly influence socioeconomic performance, with governance interventions also occurring at many of these steps. With colleagues, we've been applying this information toward proposing sustainable approaches to system level exploitation, including being able to monitor metrics like total fisheries landings for a given US region, and using that to determine system-wide exploitation rates related to things like area, total biomass, or primary production within a given ecosystem, especially since these factors set foundational limits on total available harvest and also that of particular trophic levels. These relationships with primary production can be derived from available satellite chlorophyll data, and that accessibility is becoming greater through more cost-effective platforms. And we also have pretty good estimates of landings for most US regions, and have been able to connect that to the data we have on surveyed biomass and socioeconomic metrics as well. And this information is becoming more available too. We've been applying this toward determining ecosystem overfishing thresholds, creating indices, developing ecosystem level reference points for management that relate exploitation and economics directly to those basal limits of a given ecosystem. And that information can be used when thinking about setting catch limits on total system-wide harvest and for particular functional groups like those that currently exist for total groundfish harvest up in Alaska or for forage fish in the Pacific region of the US. We've been able to evaluate progress for the Gulf and US Caribbean regions along these lines and in terms of their governance actions, but given limits on time, I'm happy to get into more details about those findings during our panel conversation. So thank you.
Thank you, Tony. Next, we have Tabitha Stadler, who is the Executive Director of Environmental Protection in the Caribbean, or EPIC. Her career spans more than 20 years and encompasses leadership positions responsible for environmental education, science, research, and policy. And with that, I will turn it over to Tabitha.
Hi, I'm happy to be representing the team of the nonprofit Environmental Protection in the Caribbean on this project that took place in 2019. It was actually conducted by our affiliate organization, the EPIC St. Martin Foundation on St. Martin, and conducted by 3 interns from the University of Amsterdam with funding from the Community Foundation of the Virgin Islands. So thanks to the partners. They did a whole range of work, but the focus of the presentation is on a household survey that they did for the Simpson Bay Lagoon. And you can see on the map there, the island of St. Martin with the French side to the north, the Dutch side to the south. That's 33.5 square miles. And the study site is the lagoon there, the Simpson Bay Lagoon, 3.5 square miles. That does include a Ramsar wetland of international significance. Yet the bay has been in documented declines for decades. The photos there show raw sewage traveling over impervious surface directly to the lagoon, algal blooms, and documented declines in corals, seagrass, and mangroves. And so, so the idea of what was the value and how was the community valuing the lagoon was of interest to affect decision making. And of course, the economy there is primarily dependent on tourism and use of the lagoon for those purposes. So the survey was door to door, 219 respondents, and embedded in that survey was a choice experiment card that looked at the willingness to pay for ecosystem services. And that piece actually was published in the Journal for Nature Conservation recently. And what they found, well, let me tell a little bit about the experiment card. Each card had 3 choices, and the third choice was status quo, business as usual, but no charge for that, which included the declines. The other choices had $2, $5, $10, or $20 amounts linked to them, and they centered around a suite of attributes or ecosystem services that included damage from storms, water quality, habitat, recreation, and stay over tourists. And what they found was significant interest and motivation to pay for improvements to the lagoon, which was really significant. And of the attributes or services preferred, the storm damage, this is on the slide there, the less storm damage, better water quality, and more habitat were ranked highest, which was interesting that recreation was not. Taking that a dollar amount of willingness to pay and then linking it to each attribute, the study was able to calculate the current cost of the lagoon at 12 million, but with improved management at much higher dollar amount. So the timing has been a little odd. In 17, we had 2 hurricanes, Irma and Maria, with significant damage. 2019 was the study, and now 2021, the pandemic. But the hope is that this will help influence management of the lagoon through the results. So, thanks.
Thank you, Tabitha. Next, we have Christopher Cusack, who works with Ocean Technology Solutions team in EDF's Oceans program and is focused on identifying, developing, and implementing technological solutions to the most pressing challenges for our global fisheries. Chris, the floor is yours.
Thank you very much. And hello, everybody. So my whole purpose of being here is really to make the case for widespread uptake of electronic reporting in the Caribbean. It may not be the sexiest thing, and it's been around for a while, but I really feel that in the current context of technological development, this is a place where everybody needs to go. So first of all, we need data. We need data because climate change is having huge impacts on our fisheries and communities. Longstanding overfishing has also had these huge impacts. Data is necessary for science-based fisheries management. And we need to use data as well. We don't just need data. We need to use data to generate value for communities, for managers, and for people who use the information. And at the same time, we also need to have communities engaged more in monitoring and management of their fisheries. So how do we do this? Well, we don't do this with paper-based systems, right? They're the easiest systems to set up, and everybody's familiar with them. But what they do is they kind of leave us at a disadvantage, and I'll tell you why I think why. First of all, we're in the middle of a digital revolution. More than half of the world's population use the internet for some kind of value, and over 3 billion people use smartphones. We have the tools, therefore, and to actually to make data collection and use more widespread. And furthermore, the time is now. And this is a necessary step for tapping into the huge potential that the digital revolution has. We start with electronic reporting, but we can imagine all the uses of technology, and we've heard about a lot of them that become enabled because of the simple digitization of data directly from the time the data is collected. And so why electronic reporting? Well, in the context of climate change, we need better scientific capacity. We don't have enough fishery scientists, as we're all familiar with. Electronic reporting can directly feed into an automatic analytics and visualization platform, which gives these tools, which gives these analyses directly to people who wouldn't normally have the capacity to do so. That's reason one. Reason two is we need faster stock assessments and faster action, from the time the data is collected to actually something actually happening on the water. This all feeds into adaptive management, and adaptive management is absolutely necessary in the context of rapidly changing ecosystems. Faster stock assessments can lead to better climate resilience of fisheries and the communities that depend on them. Going back to better community involvement, electronic reporting enables that. A cloud-based data collection system really gives analysis tools and visualization tools and, in effect, management capacity to anyone with a smartphone. That can easily be scaled across the scale of the country. And then the last reason that I'll talk about, and there's many more, but it's better fishing. We can improve fishermen's bottom lines. Near real-time data collection and analysis can feed into things like bycatch hotspot maps, and can also feed into things like traceability systems. There are a range of existing projects throughout the Caribbean, but they're kind of siloed, they're kind of spread out across the region, and including some of our work in Belize and hopefully Cuba. But we're embarking on this effort to really spread electronic reporting across the region and across Latin America, and we hope that you'll join us on this journey. Thank you.
Thank you, Chris. We will move to now to Megan Davis, who is research professor in aquaculture and stock enhancement at Florida Atlantic University's Harbor Branch Oceanographic Institute. And Megan will be talking about e-conch, e-learning for growing queen conch. Megan.
Thank you, Reed. I'm excited to share with you today a new online course that we're developing. And there were 19 authors on this presentation because this truly is a regional project. We have authors from throughout the Caribbean. You can see on the map that's on the slide here is we have a number of different partners that are throughout the Caribbean that are helping us with this development. And also some partners in the US, and we have a partner in Italy. So this project is designed to be an online course that teaches the basic skills of how to grow conch for restoration, conservation education, and sustainable seafood. And we started this concept about 18 months ago when we started to get a lot of inquiries, not only from the partners that are listed in the presentation, but also from others throughout the region. And so knowing that I personally can't be in all those places at once, I started brainstorming with the Queen Conch Lab team here at Harbor Branch on how we could start to share this information. And so that's how the online course has started to come about. There'll be 8 different modules. And the course will be a mixture of hands-on videos, presentations, and group discussions. And the participants will learn everything from the conch life cycle to aquaculture best practices, how to design and set up a conch aquaculture system, how to raise the conch from eggs to juvenile stage, how to feed them. And they'll also learn some options that exist for restoration and ranching. So I am at Florida Atlantic University and at the Harbor Branch campus. So we're partnering with the Florida Atlantic University Center for Online Learning and Continuing Education. And so the e-course is being built on a platform called Canvas. And let me see, what else can I share with you? As part of the course, what we're planning on doing for those that don't have a hatchery is putting together a kit so that those partners can have what we would call a benchtop hatchery. And then those that can participate in a hatchery, there is a hatchery in Curacao and Puerto Rico right now, and there's one planned for the Bahamas. So some of our partners will get to work in the actual hatchery as they're learning. We're hoping to begin testing the Econ course in the summer of 2022. And the partners, as I mentioned, are the partners that will be beta testing this the online course, and they'll provide feedback to us so that we can improve the course and offer it to others. And if you'd like to learn more about this course in more detail, you can watch the presentation on the GCFI platform or on YouTube. And you can also follow us on Queen Conch Lab on Instagram. Thank you.
Thank you, Megan. Okay, we will move on to Jessica Quinlan. Jessica is a PhD candidate at the Predator Ecology and Conservation Lab at Florida International University, and Jessica's presentation was using fishery contributed secondary fins to fill critical shark fisheries data gaps. Jessica, go ahead.
Hello, thank you. My research in general focuses on exploring new approaches to improve shark fisheries management, particularly in developing nations. So the talk that I gave for the conference outlined a novel method that we first tested in Belize to help developing nations overcome some of the financial and logistical challenges that they face when they're trying to collect important species and size-specific landings data from their shark fishery. So as many of you may or may not know, there's few examples of sustainably managed shark fisheries globally, and the examples that we do have are only found around developed nations. So this is often because developing nations lack the resources necessary to collect the fundamental data needed to assess their fishery. So to combat this, low-cost, accessible methods need to become available for nations with limited resources to enable them to collect these species and size-specific data that's required to assess their fishery and begin making management decisions. So the method that I'm going to talk to you about today is one of these low-cost methods and has the potential to be widely applicable. So we developed this method using secondary fins, in this case, the anal fin, which is actually the fin that I'm measuring here in this photo, to collect species and size-specific landings data. And we did this first in Belize, who has an active shark fishery. And we used the fisher-contributed secondary fins. And we got the fins from fishers who, as per condition of their license renewal, were required to remove and submit the anal fins from all the sharks that they had landed. So through this, we were able to determine that the anal fins were just as unique as the sharks themselves. And in most cases, they were able to be visually identified down to the species level. So this let us know what species were being caught and how many of each species were being caught. We then took it a step further and decided to investigate whether we could use the relationship between the anal fin length and the total length of the animal to regress the length of the shark that the submitted fins had came from. So we focused on the most common shark species, which were the Caribbean reef, the blacktip, sharpnose, and bonnethead shark. And we were able to build regressions that then enabled us to calculate the size of the sharks that were landed just by measuring the anal fins. So this resulted in length data that through length frequency histograms enabled us to determine that the Caribbean reef and the blacktip shark are being primarily landed below the length at which they mature, which is a bit of a red flag. And so now that we have the length data, we're able to further investigate this and conduct an assessment to determine whether these species are actually experiencing overfishing. So species and size-specific landings data like these are the fundamental data that managers need to be able to assess and begin managing their fishery for sustainability. And the great thing about this method is that it's low cost and it requires little training. We've actually developed an anal fin guide to help managers visually identify species in the Western Caribbean by their anal fins. And it's highly adaptive, meaning it's flexible enough to work with fisheries of different scales, different management types, and different resource availability. So it can be as simple or as complex as needed. It's not a one size fits all, which makes it accessible to more countries. So ideally, we'd like to see this as a way to standardize collection of these data, and we'd love to collaborate with more countries in the Caribbean region. Currently, we're working with Puerto Rico, Guatemala, Mexico, and Honduras to begin using this as a way to help monitor their shark fisheries. And yeah, thanks for listening.
Thank you, Jessica. Next, we'll move on to Jeffrey Burness, and I apologize if I mispronounce your last name there. Jeffrey is a co-founder of the Caribbean Cetacean Society, whose long-term vision is to create an MPA at the Caribbean scale dedicated to megafauna conservation. Jeff, the floor is yours.
Can you hear me well? Probably. So you probably know that cetaceans is a group including whale and dolphins. But maybe you don't know that whales capture 4 times more carbon dioxide than the Amazon forests. And in the Caribbean, we got so far 33 species that have been observed out of the 91 that exist in the world. And despite the fact that we are the 1/3 of the world's cetacean species, and this group play a crucial role for us, there's still an important lack of data here. We really don't know what they are. We don't know where they are, how many, and like what they do here. So if you imagine yourself as a whale, it's easy to understand that there's no way for you to recognize the different frontier between the islands. And that some place you're going to be protected and some other place you're going to be hunted. And there's a lack of geographical and temporal continuity that need to be solved if you want to like really conserve them. It means that if you're only doing project limited by time or like a small geographical scale project, it would not work without cooperation. So until last year, there was no organization that was dedicated to cetaceans conservation in the Caribbean. And this is why we created the Caribbean Cetacean Society, which is a local nonprofit NGO dedicated to this work that need to be done here. Our main objectives are to facilitate the cooperation between the islands, to actually coordinate large-scale scientific research, to support a better conservation policy, and to educate the local population. And we work actually as a network with different focal points in the different islands that can help us. So this year, we accomplished the largest survey in the lesser hunted with the Tiwel-Onu expedition that actually some people here participate on. So we've done boat transect during 4 months from Anguilla to Grenada in all the Caribbean lesser Antilles. And in order to actually understand more about the population structure of the sperm whale and the distribution of the different social clan. So sperm whales speak different languages, and this is according to the different culture they belong to. This is why we actually use passive acoustic monitoring with a towed hydrophone array and also photo identification. So as a result, we actually encountered 16 different species, 29 families of sperm whale, more than 10,000 individuals of cetaceans. And despite the COVID situation, we actually were able to do capacity building to train local people in the different islands. So we're planning to reconduct this kind of large-scale project every year. And next year, our focus is going to be on humpback whales from March to May. And we'll actually invite all the local scientists interested to reach out to actually participate in those surveys.
Thank you, Jeffrey. And our last panelist is Jaime Bolaños, who is a Colombian and Venezuelan marine biologist with a 30-year career focused on cetacean research and conservation in Venezuela and the wider Caribbean region. And Jaime is going to talk about killer whales. Jaime,
Hi.
Start.
Buenas tardes para todos. Voy a exponer en español. Muchas gracias por la oportunidad de participar aquí con este trabajo acerca de la orca en la región del Gran Caribe, donde revisamos la ecología, la explotación e interacciones con pesquerías. Este proyecto es a nombre de mis coautores de la iniciativa informal Proyecto Orcas del Caribe o Caribbean White Orca Project. Es una iniciativa que surgió hace 10 años mediante alianza entre investigadores de Aruba, Venezuela y Holanda, fundamentalmente para la publicación de registros ocasionales de orca en nuestras aguas. En el proceso de recopilar la información para publicación, nos dimos cuenta, o se fueron incorporando investigadores de otras regiones que también tenían datos de orcas a nivel de todo el Gran Caribe. De tal manera que se reflejó un esfuerzo cooperativo e interinstitucional mediante el cual logramos producir la primera revisión exhaustiva de la orca en el Caribe Para ese momento, en una publicación del 2014, contábamos con 176 registros de orcas en el Caribe, que definitivamente la posicionaban como una especie caribeña. Nos dimos cuenta de que ese enfoque colaborativo e interinstitucional era útil también para revisar otras especies y producir información acerca de cetáceos del Caribe, e incorporamos a la ballena minke y otras especies. Entonces, con referencia a la orca, le dimos continuidad al proyecto. En los actuales momentos contamos con 332 registros que abarcan desde 1851 hasta este año, y hemos producido información acerca de la ecología, de la distribución espacial, las interacciones con pesquerías y recopilar información acerca de la operación de caza comercial en San Vicente y las Granadinas. Como resultados principales tenemos que las orcas están presentes en el Caribe durante todo el año. No hay diferencia estadística ni por mes ni por temporada, pero sí hay una tendencia a mayor cantidad de registros en los meses cálidos. La mayor parte de los registros proviene de proyectos de investigación científica, pero las iniciativas de ciencia ciudadana están aportando alrededor de un 40% de información. Respecto a la dieta, hemos encontrado que la dieta de las orcas en el Caribe está basada en más del 60% en cetáceos odontocetos y aproximadamente un poco menos del 20% en tiburones. Y para finalizar, entre las principales amenazas que hemos detectado están la operación de caza no regulada en San Vicente y las Granadinas y los enmayamientos. Muchas gracias.
Thank you, Jaime. Okay, so that concludes the panelist overviews. We will now open the session up for questions. Again, please use the Q&A button at the bottom center of the Zoom window, and those can be directed to specific panelists or general questions. Don't be shy. Well, I will throw one out that I guess it is probably more appropriate for those speaking on the innovation side, but I think it certainly pertains to the one on megafauna, excuse me. And so I'll just toss it out there for discussion. So what do y'all see, particularly on the innovation side, what do y'all see as your biggest barriers to success for your particular efforts? And you can't use funding, that's the universal one. So what's the biggest barrier? And Tony, if you have some thoughts, maybe get the conversation going.
Sure. Well, yeah, definitely thinking about advancing ecosystem-based fisheries management. And we've been looking at this in the Gulf of Mexico and US Caribbean, which are both known as kind of data limited regions. So there are certain limitations right now. There are quite a number of unassessed stocks. There's historical overfishing in these regions, challenges with getting accurate commercial and recreational landings. There's also unknown biomass estimates for key functional groups in the US Caribbean. But despite that, we're still seeing a lot of progress in both geographies in developing broader management approaches. There are fishery ecosystem plans, for example, current island-based fishery management plans for the US Caribbean that are forthcoming or currently in place. And this is helping us get a greater understanding of their socio-ecological systems. There are a lot of other governance frameworks that are being put into effect right now to overcome some of these limitations. But certainly data limitations right now is a big key thing, not just for the Gulf and Caribbean, certainly applicable to many US regions. And those are things that we're continuing to advance forward on, even with some some recent released EBFM implementation plans that now exist for all of our US regions. So we're still optimistic, certainly a lot of work to do, but those are some challenges that we're working to overcome at the moment.
Thanks, Tony. Any others want to talk about challenges y'all see in your next steps or long term?
I think the biggest barrier for technology implementation across the world, and this is not just in the Caribbean, is the fact that the capacity of managers is really limited. And the ways that managers use to collect information and use information are kind of limited to fishery management plans, as Tony was just talking about, which are really critical. But we feel that we need to be more inclusive of the stakeholders of the fishing communities and the fishermen themselves. And so finding ways to not just collect information for fishery management plan purposes, but to both integrate fishermen into the management process, but also find ways that we can use that information in other ways that increases their bottom line. We feel that that can really provide the incentives needed and the momentum needed to really get data collection and use at the scale that we need it to be.
Thank you, Chris. Any others on challenges? Okay, we did have a question that was submitted for Jessica on her pre-recorded talk. And so this is for Jessica. How did you convince fishers to send the anal fins to you? Was there a kind of gratification or something? I've been working and dealing with fishers, and it is not easy to get them involved.
Great question. So thank you for that. Yeah, it can be quite difficult. We initially conducted a pilot study for this project where we did incentivize fishers we paid them a Belizean dollar per anal fin, and chose that because we felt it was enough that it would incentivize them to go through the effort of setting the fin aside, but not enough to incentivize increasing capture of sharks. And that was just to sort of understand if we could use the anal fins as a way to identify the sharks. And then since then, we're fortunate in Belize, we have a unique case that Belize has a management strategy in place that their fishers are licensed, and it just became a requirement for them to hand those in. But as far as when going to areas where you don't have such an advanced management plan where fishers are licensed, you can look for ways to incentivize them. And it doesn't always have to be money, right? It can be, whatever commodity is of value to them. Sometimes it's gas. I've worked with fishers where I've brought them, tanks of gas for their boats to have access to their landings and things like that. I know of one company or one NGO that incentivizes with offering scholarships for students. If you can come in and sort of get access to their fisheries. So there's definitely ways. The best way, of course, is to build those relationships. And this kind of circles back to the barriers question that you had asked, and what Chris touched upon was building those relationships with fishers are just paramount to any of this, because once you have a relationship with them, yes, of course, you still want to incentivize them, and you want it to be a two-way street where you're offering and not just taking data as well, but offering something as well. But building that foundational relationship is really what opens the door, and sometimes that's not easy and it just takes time, or you won't be able to do it with all fishers. So yeah, there's no straightforward answer, unfortunately, to answer that, but it can be challenging, but there's certainly ways around it. So.
Thank you, Jessica. I have a question for Jeff. Can you talk about the process of engaging with local departments within the region to get them involved with your work? Talk more about your plans to expand your work. Are you simply expanding to include other species, or are you expanding the geographic area?
So it's completely different between the different islands. So each island is going to have another governments, and from the French government to the Dutch one to the OCS government, it will be completely different to get the permit, to get people engaged, to actually get whale watcher or actually fishermen engaged too. So there's not only one answer, and it's just keep talking to people, meeting them, going in the island, show that you're present and you're not from outside. And I think it's pretty important. So we're not going to expand to other species. We're going to stay on cetaceans. And I think there's already a good job that have been done for other species. And concerning the actual range, the geographical range, for now, our funding only sufficient to actually do the lesser ante, but we would like to actually extend to do more and more. and to probably do until Dominican Republic next year if possible.
Thank you. We have a question for Jaime. Can you talk more about the impact of fishing gear on killer whales? Are there specific gear types that are affecting the species more?
Ok, en realidad la proporción de avistamientos que en lo que hemos detectado interacciones con artes pesqueras es relativamente baja. La interacción con actividades antrópicas la hemos encontrado aproximadamente en el 20% de los casos. Eso incluye interacciones con dispositivos agregadores de peces, con anzuelos e incluso hasta orcas aparentemente alimentándose de peces arrojados por pescadores. Las relaciones con otras especies se han documentado aproximadamente en un 10% de los casos. Incluyen mamíferos marinos, tortugas y peces. El caso de la operación de caza comercial en San Vicente y las Granadinas es bastante puntual, pero durante muchos años fue la mayor fuente de información sobre la presencia de orcas en el Caribe. Así que la respuesta concreta sería que las interacciones de orcas con actividades antrópicas están alrededor del 20% de los registros que tenemos disponibles.
Great, thank you, Jaime. One for Tabitha. So you did an initial economic valuation studies. What's the next steps moving forward?
Yeah, well, I mean, our main goal is to motivate some changes in the management of the lagoon. I will note that I think there was a degree of surprise around the results. It's pretty typical to say, it's expensive and people won't pay extra for some kind of environmental improvement, but yet the results just really resoundingly said the opposite. And I didn't even have the chance to note that beyond the choice card, willingness to pay, the actual survey itself asked some very direct questions, such as, had you seen changes in the lagoon since you arrived or in the last 10 years? And if so, what were they? And then if so, what would you do about them? And so there was also just survey question wise, willingness to invest in outreach and education and research, things like that. So we have it has worked for us to use this information to get grants. So I guess third party sort of groups tend to really find the research valid and compelling in terms of wanting to fund projects. But we would like to see some government work on a higher level. For example, we asked about 3 management scenarios. One was mangrove restoration, which is something that we do as a nonprofit, and one was in putting in a sewage treatment plant, which would obviously be more of a government intervention. So I think our thought is to try to work more closely with some of the government departments to see if there was a way to kind of leverage that information. We are concerned about doing a follow-up survey, though. I mentioned quickly at the end that the timing was so odd because with the pandemic, so shortly after the survey, economically, that may completely change people's willingness to pay, although it shouldn't necessarily change their feelings about the importance of restoring the lagoon. So we need to do some messaging around it. And my last point is the messaging actually is really important and kind of an outcome that we hadn't fully anticipated, which is a lot of messaging that I've seen has been around recreation. If you noted that recreation wasn't considered one of the primary values. So normally you would say, oh, let's make it swimmable and fishable in 10 years, right? Let's have a campaign and implement a series of steps. So we would have to pick steps that were, messages that was more perhaps around the storm damage or the economy. So yeah, possibly a follow-up survey just in light of the pandemic and the changes in the economy. And then 2, just messaging and trying to use that as a point to create a management plan, essentially, that gets both government and private sector and nonprofit buy-in.
Great, thank you. I have a question for Chris. The potential scientific and management benefits for electronic reporting are obvious. However, fishers can be reluctant to report catches accurately with concerns that reporting may lead to sanctions such as reduced fishing quota. How do you persuade fishers to get on board with electronic reporting, and how do you keep them involved in reporting accurately?
That's a fantastic question. Thanks to whoever asked that. And there is no real clear answer. I think, it's all about the incentive structure that fishermen face. And when fishermen are not part of the management system, when they don't see how their actions are reflected in management actions, then they tend to feel disenfranchised. So I think the answer lies in incorporating fishermen into understanding what performance indicators and harvest control rules are and how their actions affect them. I think education and capacity building is another important component. I think the carrot that I was talking about, where we try and improve fishermen's bottom lines, also has a really important role to play, but not at the detriment to the capacity building and inclusion processes that need to happen as well. Does anybody else on the panel have any thoughts on that? That was a great question.
Yeah, please weigh in as you like, and I can reread the question if I need to.
I was just going to say, I'm so sorry because I was fielding a question in the chat and I think I missed the question, so I apologize.
Hold on. Okay. So it's talking about the electronic reporting, scientific and management benefits, and how those are obvious, but it's fishers being reluctant to report catches accurately because of the potential outcomes in terms of reduced fishing quota. So how do you convince fishers to get on board with what you're doing? How do you keep them involved, and how do you ensure, for this particular question, they're reporting accurately?
Okay, yeah, I think at the core of it, it circles back to also the trust building and the relationships they have with these communities. Whether they're doing something illegal or not, I think that's a huge factor. And then as far as reporting or fear of reporting, and that, maybe what they're reporting will contribute to lowering quotas or something like that, because I think that's sort of the gist of what this person's asking is, I think what Chris had mentioned also is education, having them understand that lowering a quota isn't a punishment. It's actually a way, explain to them how the process works in the sense that if we take a break from fishing something, it'll actually help them in the future. Talk about ways that if that affects their income, make them a part of the conversation essentially. And the more they become part of the conversation and the more they understand the efforts and the work and the results and the hopeful outcomes of everything that we're doing, I think they're more inclined to want to work with us and understand that we're not here to do research and cut off fishing and make fishing stop, or claim that everybody's overfishing, that we're on their side. We want to rebuild stocks so that they can have livelihoods that will last and that are sustainable. And I think that's just a conversation, and trust and capacity building and education is a huge part of that as well. And then there's always alternative livelihoods that can help offset some of these things if they are fearful that their jobs are at stake or things like that. So.
Thank you, Jessica. Any others want to weigh in on the trust question?
Yeah, thank you. Certainly, and we've been seeing this many locations, that stakeholders need to really see that the information that they're providing or working together with managers and scientists, it's really having real outcomes. And it's really, we're taking that information that they're providing and doing something with it. There are many fishermen who I've talked to throughout the Gulf of Mexico, for example, who they want to provide otoliths. They really do want to participate in cooperative research, and in many other regions too. But they need to understand that, what is this ultimately producing for us as the fishing community? When we were doing evaluations of how Hurricane Harvey was affecting the Texas community, especially shrimpers and fishermen, I heard concerns of, hey, you know, we are providing these kinds of data, we want to work together, and we're not sure how it's ultimately getting incorporated. And there's even concerns that that information that we are provided is going to lead to negative effects on us. But I agree, there needs to be more education, there needs to be more direct engagement, there needs to be more understanding of how we benefit from those who are on the water, even for things like EBFM. Lots of the conversations have been, you know, we need to go to these resource holders who are on the water, who are understanding, hey, these are the big things that are affecting us right now. There have been some great workshops that have been done off the Gulf Coast of Florida after the red tide events and hypoxia events to really get fishermen on the water, collecting environmental data, and helping us enhance our models, enhance our understanding of how these effects are, of how these environmental phenomena are affecting fisheries, or affecting our stakeholders, or affecting fishermen on the water, and creating that trust. And that's working its way up into some of these even broader pie in the sky kind of management approaches and things that we really want to get at for understanding the full system. So yes, there needs to be more of that trust. There needs to be direct understanding and communication to the stakeholders of what their data is creating, how it's being applied, and take that back to continue fostering those relationships. And I've been very pleased with what we're seeing with our cooperative research programs, and really even trying to get at higher resolution information for continuing our awareness of how these systems are responding to all sorts of future pressures.
Reed, I have a few comments I'd like to make in regards to working with the fishers in our aquaculture project in Puerto Rico. Yeah, thank you. In the last session, I presented our work with the Puerto Rican fishers at the Fishing Association in Naguabo. And my colleague Raimundo Espinoza, the executive director of Conservación Conciencia, is been working with this fishing association for about 6 or 7 years on a number of different projects, and had started to build up, or has built up, an amazing rapport with the fishers there. So that when we came in with the idea of having an aquaculture facility right there amongst the fishing association, like right there on their grounds, and having the fishers involved as a diversified income, so not an alternative, but a diversified. And so when they go out and fish, they for conch, they look for the conch eggs, so they know exactly where to go, and they bring back the eggs. We certainly compensate them for their time on the water and finding eggs, and their expertise on the water and what they know. And the other side of it was that we needed to make sure that when we met with the fishers, that they didn't feel like this aquaculture facility was going to be a threat to their livelihood, that they could still fish conch and still bring in conch, and this wasn't going to be a competition for their resource. Instead, we work in unison with the fishers, and they'll be the ones to help us determine where to do the restoration and determine where to do the grow out of the sustainable seafood, and they'll be the ones to take care of the conch in the field. So I just wanted to mention that this is a opportunity that we're working directly with the fishers and how we built up the trust using aquaculture in their facility.
I was going
to that's a great point about the perception of aquaculture being competitive with wild fisheries. It's something I think that gets overlooked, but it sounds like y'all are doing a good job of communicating that it's mutually beneficial. Tabitha, did you want to weigh in?
Yes, I think it's a little bit of a tangent, but it's related in that there are not all that many studies about, I mean, there's some on like sort of participatory decision-making and management, but in terms of watershed decision-making, I was involved with a study that I thought would be relevant in terms of trying to, in terms of management, which is just that in interviewing lots of people, that sort of word of mouth and who you know, and trusting your trusted neighbor or other people is important to go beyond the target audience of just the fishers, and to be having strategic conversations with people at other sort of parts of the community, because as we know, they're small communities and everybody knows everybody, and so we've had a lot of luck in doing kind of like something with kids and then something with decision makers and realizing that different professions interact with these other professions. And part of that study that I'm referring to also noted that people who like have a profession that relates to water tend to be respected among their peers, even though they might not be a direct fisher. They might be at a restaurant and buy fish, or they might be in some related marine industries business. But by sort of educating beyond the scope of the immediate target audience, we found that had many trickle down benefits in kind of creating, and again, what that study said was there's a little bit of a group think going on. That is, once everybody sort of gets on the same page that, oh, these are good people, this is a good idea, they're doing good work, then that word spreads, and that seemed to have significant positive benefits. It takes extra effort and kind of beyond the comfort zone of the work that we were doing, but it seemed to really pay off in better management, more acceptance on a broader community level of management changes, essentially. Does that make sense? Do you know what I'm getting at? Yeah.
Yes, it does. Thank you. So Tony posted a question for Jessica and Chris. Are there particular areas throughout Central America and the Caribbean where there are complementary efforts using these kind of technologies or approaches, or where they're becoming more popular?
I'm happy to take a first stab, Jessica. So in terms of electronic reporting, efforts across the region, as I said, are quite spread out and siloed, but there are several use cases. For example, in Belize, Shell Catches, which is a technology company, they have an e-reporting app that's been used that integrates directly into a traceability app called Fresca Pesca, which then presumably offers fisherfolk a premium over the market price. In Mexico, an app was developed called Pesca Data that helps fishers and fishing organizations offer their products and services in the marketplace, share knowledge, and seek solutions to common problems, but also record catch and fishing effort, expenses, product prices, and more. And that's just a couple of examples. One area that we're working in Belize, we're working with a company called Veracatch, and their application is called Fisheries App. We're excited to implement that in the Turnaf Atoll Marine Reserve. But the goal of the project is to really think about how we can use these proof points or these demonstrations that apps can increase the efficiency of the process and bring benefits, to electronic reporting at the scale of the entire country. Fisheries App has a sister application called Know Your Fish, which provides the ability to trace via lot numbers and doesn't require a physical product identifier, which is often a barrier in traceability systems. And so those kinds of ecosystems where you're collecting information, but then you're using it in multiple different ways and for different purposes, and allowing people to come in and create value from that information, I believe is the future.
Thank you, Chris. Jessica, so there are particular areas throughout Central America and the Caribbean where there are complementary efforts using these kind of technology or approaches. Are you aware of any?
I'm not. So I'm actually out of the tech loop, so to speak. I'm not really up on. So I think Chris definitely would be the best person to field that question. And that I learned something too from Chris. So that was great. Because I don't, know much about the tech applications.
Okay, another chat question, again for Chris. When it comes to digitizing data collection and reporting, how easy has this been for implementation, and how has this been received? Can you give more detail from the Caribbean perspective, since there's often limiting existing capacity within fisheries departments?
Sorry, can you ask the first part of that question again?
When it comes to digitizing data collection and reporting, how easy has this been for implementation, and how has this been received?
Right. So I should say that we are starting off our journey in the Caribbean and Central America region. We have a project ongoing in Chile that's actually just been kicked off now, but we have been working in Indonesia. EDF has a country team there, and we work in 3 separate regions. 2 of the regions are we work in a crab fishery, the blue swimming crab fishery, and the 3rd region we work in a multi-species fishery. We've implemented Fisheries app in that region, and I guess we've had a bit of a leg up because we have existing partnerships with government, university, and local stakeholders, and so the stage has been set for our implementation of electronic reporting. When we first implemented the app, all of our enumerators or community observers who would collect the information had their own smartphones, and so we just loaded the app onto the smartphones, and we paid them a monthly fee to collect the information, and that happened. That went pretty well for about 6 months or so, but then it became clear, after continued dialogue, that enumerators were worried about dropping their phones in the water or getting crab slime on them, and so the project kind of slowed down until we decided to buy smartphones specifically for that use. And in Indonesia, the smartphones were about less than $100 each. And since then, we have collected thousands of records easily. All the records get transmitted via cell data onto a central database. It's connected to an analytics and visualization platform where fishery managers can actually go in and see what's happening day by day. And so there are always implementation challenges when it comes to tech, but in my experience, they're mostly surmountable.
And the second part of that question was, can you give more detail from the Caribbean perspective, since there's often limiting existing capacity within fisheries departments?
Yeah, so I tried to answer that at the beginning, but we are just at the beginning of our journey into the Caribbean space, and we're kicking off projects in Belize. We're talking with partners in Cuba to try and kick off a project, and we have kicked off a project in Chile, which is not the region, but I'm happy to give an update in maybe a year or so to how those implementations went.
Thank you. Do we have any questions among panelists that y'all would like to ask one another? That's the one that Tony had posted in the chat box. Okay, I have a question for Megan. So your eConch app, you're going to try to reach more people through technology and the virtual world, which obviously has greatly expanded over the last several months. So if that is hugely successful, what is your next step for the actual boots on the ground training that some of these people may need to do? Is it a plan for you and your staff to go to them to try to centralize training somewhere? How would you handle those logistics?
Yeah, thanks for that question, Reed. So part of the idea will be to have the virtual platform for training on techniques to work with queen conch, but also to make the other hatcheries available as training centers, especially Puerto Rico as a centralized area in the Caribbean, having workshops there, having people come. We already do that. We've had people from Jamaica and Bahamas visiting us to learn more. So I think that's always going to be a need to be able to go to a place and actually have some time with the aquaculture systems and with the species itself. I'm excited to see how the kits work, because I think for probably $3,000 or $4,000, meaning a microscope and some small beakers and glassware, that just teaching the behavior of the animal and understanding the behavior of the animal can be done in sort of like a benchtop type of hatchery. So I'm excited to see how that will work. I've certainly been growing conch now for over 40 years, and I've been everything from commercial aquaculture down to experimental small scale. And so I'm excited to see how we can put those sort of lessons in place. I'm also at that stage of my career where I really want to leave the knowledge as a legacy, so that there'll be more ambassadors and more people trained to be able to go out into the Caribbean and teach conch aquaculture. It's such an important species in the region, and it's just important for more people to understand it and to know it. And just speaking, I know I'm going on a tangent a little bit, but I was just reflecting on the conversations with the rest of the panelists here. Building that trust with the fishers, what I've been seeing is that aquaculture has been an amazing tool to teach about the life cycle of a species. And so we're starting to see, especially in the Bahamas and Puerto Rico recently, I've been starting to see how the fishers starting to understand a little bit more about the biology and why it would take 4 years to be able to wait to fish a conch, so that it's had a reproductive cycle already. And just about why the conch in their nation is important for other nations, because it's a planktonic veliger that's drifting. And so I think that the e-conch program will not only be beneficial for anyone that wants to learn about conch, but it's sort of those concepts can be used by those that learn within their own community. And so we're also hoping that there'll be like this ripple effect from that as well. So thank you for the opportunity to talk about this a little more.
Thank you, Megan. Any more questions or thoughts from the panelists? I think I'm preaching to the choir, particularly with Gulf and Caribbean Fisheries Institute. But when, from my perspective, when dealing with folks within the fisheries, the fishers, the anglers, I think oftentimes you're particularly coming from a perspective of research or academia. It's having the individuals understand that you're there to learn from them. You're not there to try to teach them. It's such a big component. And I've told everybody who has come through my ranks, is if they're out in the field working, if they're interacting with folks within the fishing community, the biggest mistake you could make is to think that you know more about what's going on in the water because you went to school for 2 more years or 4 more years. The individuals out there working those boats know more about, looking out at the Mississippi Sound right here, know more about what's going on right there than any of us in science ever will, because they spend their life on the water. And so I think that that attitude of please teach me, I'm here to learn from you, really helps to loosen up the tightness that may be involved in those initial relationships. So with that, with me ending my little bit of rambling there, we will go ahead and close this session. Again, like to thank our panelists for their presentations, the information they've shared with us today. I also like to give another big thank to Fadia, Alejandro, Bob, GCFI board for all their work in organizing this virtual conference. I think that the platforms worked well. We're obviously well trained in a virtual world, but I'm sure I speak for most of the attendees when I say that I very much look forward to seeing everyone in person. So with that, I will turn it back over to you, Fadia.
Thank you, Reed, and thank you again to all our panelists today and also to our moderator, Reed.